Well, yeah, some of you already know me as a science communication coordinator for this cost action. But as Nina told, I'm also the vice director of our pole of ID+, that it's a research center that we share with other institutions. We had two main poles. There is the main, or two founder poles. There is the pole of Aveiro and the University of Porto. We have two other institutions. That is a polytechnical school in Barcelos. It's a small city, not small, medium city on the north of Portugal, and another polytechnical school also from Porto. So we are one of the biggest research units in design in Portugal. In Aveiro, we have this focus on interdisciplinarity, and we'd like to work with many different scientific fields. And so in this context, DESIGNAE, it's almost our most recent and most visible vision or output of this vision for our research unity. So, welcome again. I wish you a wonderful time here in Aveiro, and I'll be with you during this day. So if you have any complaint, anything, any question, I live in Aveiro, so I'm very informed about any solution you need in our city. So have a wonderful time, and let's start with work.
Thank you, Gonçalo. All right, so many of you already know, I'm just going to give a very brief overview about this cost actions. What is DESIGNAE? What do we intend to do? So DESIGNAE is a research network which aims to advance nature-centered biodesign for regenerative solutions. We are aiming towards different fields, namely architecture, healthcare, and emerging fields of technology. We want to integrate different research areas, namely biology, material sciences, engineering, design, and law. We do have lawyers in this cost action. We also want to address ethical and legal challenges of working with living systems, namely in terms of governance, biodiversity rights, and responsible innovation, which are the next challenges in design, and also foster interdisciplinary collaboration. We have a couple of challenges to achieve this. So the first one is, how do we transcend these human-centered concerns and mechanistic mindset in bio-inspired sciences and design? How do we integrate ecosystem complexity into our activity? How do we balance this environmental viability, technical feasibility, and economic scalability in biodesign, and how to navigate bioethics, governance, and regulation in this field? So there are many ways of looking into this in this topic. We have this initial framework, but it's very discussable. And it really depends on your disciplinary background. We are seeing this as an evolution from design inspired by nature towards designing with nature. So how do we collaborate and use natural systems or their materials to create new structures? Designing for nature, how do we restore the ecosystems which support biodiversity? And designing as nature is this intertwine between natural and built worlds. I'm the chair of this cost action. Pierre, who is also here today, is the vice chair. We are both what they call young researchers, so anyone below 40 years old. I didn't make this criteria. And Pierre is from the Regenerative Biodesign Lab, is also the co-founder of Urban Reef, And so it's from the Netherlands, and I'm currently a tenure researcher at the University of Aveiro. My background is mainly in product and service systems design. We also operate across 6 working groups. So very quickly, so the first one aims to bridge this life sciences with design, creating a shared terminology amongst our different disciplinary areas, and also try to map which type of cases and applications there are in the members of this actions of this action, because we're already 200 plus members. The second work group is dedicated to biotechnologies and materials. How do we develop, reproduce, scale these materials while also informing legal and design frameworks? The third work group is dedicated to computation, AI, and modeling in biodesign. So how do we simulate biological processes and enhance our predictive capabilities? The fourth work group aims to bridge theory and practice. So it's a very hands-on work group to develop concepts, approaches, models, prototypes in different sectors, so architecture, healthcare, or other emerging contexts. The fifth is about evaluating biodesign feasibility, talking about ethics, regulation, societal impact, and also discuss about the educational implications of our network. And the sixth work group is aiming towards coordinating outreach activities, education, stakeholder engagement in both academic and non-academic entities. So this action is not only aimed towards researchers only, it's also for practitioners. I would like to acknowledge publicly the many people that are involved here. So we do have a triangular leadership across the 6 work groups. So I would like to give a very warm thank you to Eleni Charupia, Susana Gonzaga, to Cristiano Pedroso Rosado in work group 1. You already know them, or you'll meet them today or tomorrow. In work group 2, we have Elisa Biala, Victoria Outram, and Ruben Silva. Thank you to the 3 of you. Work group 3, Renata, Branimir, and Ivana. Also, thank you very much for all the work you put in this action. Work group 4, we have Deria, Yelena, and Adrien. Thank you for putting also the energy for this work group. Work group 5, we have Pratik, Gemma, and Pascal. Thank you. And work group 6, we have Antonella and João, which is not with us today. We also have Susanna couldn't be with us, and Ivana as well, and Deria due to traveling complications, but most of us are here, and you will meet personally these leaders today and tomorrow. As for additional leadership, so we have Gonçalo Gomes, who you already met, who's the Grant Communication Coordinator, Luisa Reissig from the University of Humboldt, Germany, who's the Grant Award Coordinator, and Stefano Brossese from Torino, Italy, and Network Balance Coordinator. So some statistics very quickly. We have 29 cost member countries in this action represented in the management committee, from which 18 are ITC countries. So ITC countries stands for Inclusiveness Target Countries. We are currently 212 members, plus 74 submitted applications still to be reviewed. And there's a nice distribution across the work groups with higher emphasis in work group 2, so materials and technologies, and work group 4, which is connected to applications. So for the ITC representation, Portugal, despite coordinating this action, is not the country who has most members subscribed. It's Turkey that takes the cake. So 56 members, and then Portugal with 31 members, and then you have other countries with 2, 3 other members. And for non-ITCs, we have higher representation of Spain, followed by Italy, Netherlands, Denmark. As for seniority, we also have a very good percentage of young researchers, what they call young researchers, so 42%, and a very nice also gender balance, so we are at 50-50. For this first DESIGNAE conference, we received in 2 weeks' time, so it was the time we had to launch this call, and we did it in a record time, I believe. So we did receive 60 applications, from which 30 were approved for funding. All of them were, all of them were really nice. But we could only fund as much. So, and today we have 29 presentations during the 25th. As for the representation again, so young researchers in this conference, so we have higher percentage of young researchers here. So thank you, Stefano, for providing these graphs, and also higher representations of ITC members, followed also by a nice distribution of gender balance and higher representation of female members. As for the program today, so we have a 2 days program. The program is listed at the front of the auditorium. We have 2 panel sessions during the morning, and the rest of the panels are in the afternoon session, so it's going to be a bit longer. We have 2 coffee breaks along the day. The lunch break will take an hour at most. Some of you already registered either in the vegetarian restaurants, other in the Portuguese buffet. So I will ask you to please, if you don't recall which one, there's the list of reservations in the front desk so that you can go and check it out. As for tomorrow, so tomorrow is a bit different, so it's not going to be here. We're going to be in 2 locations. The first one is Ecomar, where we'll have the management committee meeting. There are 2 departure times for a bus at 9:30 for MC members plus core group members, and another one at 11 for other invited members. I would ask to please arrive a bit earlier so that this is the departure time. Okay, so the bus leaves at 9:30. Yes, so for people coming in the second hour, so at 11, they will be driven from the University of Aveiro to Ecomar. We will have a visit at Ecomar, which is the Marine Research Center, which is very nice. So it's going to be a quick overview made by Ruben Silva, who is here with us today as well. And then we will go, after this visit, we will go to Caixa Criativo to have our lunch there. The bus is taken at the Reitoria, which is the front building. So we are at DECA, which is here, and the Reitoria building is on the other side, so it's that way, okay? So that everyone knows where to go. This is a long ride, so 10 minutes here, 10 minutes there. Please, again, don't arrive late, otherwise you'll miss the bus and then we don't have a solution for you. There's no plan B. So yeah, so that's it. As for your credentials, if you have not already done so, please sign your presence. Your signature is required at the registration deck. You have a credential which has the Wi-Fi password. You have also tickets for your meals for the lunch on the 25th, dinner on the 25th if you have the reservation, and then lunch on the 26th if you already also had this had the reservations. For people that didn't pay, that's okay. You can pay on location, that's fine, okay? For the vegetarian restaurant, please follow Pierre. You'll go to the vegetarian restaurant. And for the Portuguese buffet, you follow Gonçalo, all right? Okay. Okay, again, so if you don't, if you're unsure about what you have ordered in these different days, you can also check again the front desk and see which meals you have reserved. Yes, and for people that are not subscribed to the working group sessions, but that really want to go, please let us know, okay? Again, front desk, we have a list of work group distribution for the afternoon on the 26th tomorrow. If you want to be a part of one of the work groups, let us know, okay? We will integrate you. Any news about the event of today and tomorrow will be communicated via WhatsApp. So please, I will not be, it's going to be complicated for me to answer to any emails that you send me at this point. So please, any questions that you have, just WhatsApp, all right? So we are all there. We can all answer to your questions, any doubts. If you're lost, let us know through this link. Also, a warm thank you for the local organizing committee and the volunteers that made all of this possible, to accommodate all of you and to have very nice meals and coffee breaks. Yes. I'm not going to go over too much about this cost action. So you already know that this is a 4-year cost action. So we have a lot of time together. The first year is mostly dedicated to work group 1, 2, and 3, although we are already starting the other work groups at the same time. So we will have lots of different moments, activities that we will be together to discuss about topics of DESIGNAE. Each of the work groups has specific expected outcomes that you can check in the Memorandum of Understanding of the Action. And there's also other opportunities for you if you are a young researcher, if you're a PhD student, if you are a senior researcher. So we have mobility funds to attend meetings, to attend working group sessions. We also have funds for short-term scientific missions, so from 5 days to, I think it's, up to 90 days if you are an early career researcher, to promote skill development, data sharing across institutions. We will have, not in the first year, but later on, training schools that you will be able to attend, to be a part of, to co-organize if you want to as well, and conference grants. So anything, if you have a specific paper that is connected to the topic of DESIGNAE, you can also apply to have access to this funding. The calls for all of this are expected to open in April 2026. So tomorrow we will discuss a bit more with the management committee about the processes and the criteria of selection, etc. But please stay tuned in the website about these specific opportunities for you. And without further ado, because we're already, oh, okay, we're not late, okay. I would call in Eleni and Pedro for moderating the first session, which is Bridging Life Sciences and Design. So please. Just a quick instruction for the in situ people and for our YouTube online audience. We do have this QR code that you are welcome to use to ask any questions that you feel like asking to the presenters of this session. Yeah, go ahead.
Okay. I guess we should also call the people who will present in our panel. Yes. So Emmanouela, for sure, I know is here. Yes. Who else? If someone's missing, what do we do? Okay.
So we have Pedro Coelho.
Pedro, for example. Yes.
Carmen, Ebru, Emmanouela, and Maria Clara Gomes.
Very nice. So hopefully everyone is here. Hello. So you can sit, please.
So they have their presentations. I'm not sure if they should sit here because they won't see the presentations. Okay. They can sit over there while, you know, and if you want to have them at the end or in between.
Okay, so you want to see outside, and then we will call each one. Okay.
They can sit at the
front row. Pedro is presenting first. Yes, it's you. Please. So your presentation is here? Yes. Amazing. Here you go.
Thank you. Good morning. My name is Pedro Coelho. I'm a biologist and I work in the biology department of the University of Aveiro. And today I'll be talking a little bit about the subtidal seagrass nature-based solutions that we are developing in the Rio de Aveiro for the conservation, restoration, and the system management. This work is being developed under a LIFE project, Life Seagrass Rewild, which started in 2023. It will go on for 7 years, up to 2030. The University of Aveiro is the coordinating beneficiary. We have other partners, other research institutes associated, and also environmental consultancy agencies, NGOs, and also the regulatory administration associated. This project evolves around the concept of seagrass mariculture. For those of you who don't know, seagrasses are a very important habitat. It's an engineering ecosystem which adapts and transforms the ecosystem, the habitat. And it's very characteristic of and very associated with Aveiro. Over time, due to various issues, the subtidal seagrasses of the system completely disappeared, and the idea is to bring them back. But because there aren't any, we have to source them elsewhere. And a little bit as for farming, for agriculture, we will farm seagrasses under controlled conditions in tanks, former salt pans that are being adapted for this purpose. And then, as soon as we have a robust population, we will transport them and transplant them into the system for the restoration purposes. The idea is to do this with the stakeholders, with the people. We started talking to them from the onset of the project. Of course, through the co-development of solutions, but also to engage them and to get them on board as volunteers, because this project has it will be very demanding in terms of manpower, and the idea is to work through citizen science initiatives to have the population on board and have them also participate in the restoration of the system. Then we have a social awareness component. This is the part that the NGO will participate, because they have implemented a similar network in Sado, a system further to the south. And the idea is to have local fishermen and fisherwomen trained so that they are awareness agents for the project, and also seagrass monitors, and this will provide them an alternative income when fisheries are closed in the system, for example. So this is also a way to give back to the people that live from the system. And because we want to have these actions with the people, but also for the people, we have the idea to develop this nature-based solution to maintain the channel navigability. The Rio de Aveiro is a very dynamic system, which, because there's a lot of sediment transport, there is the need to periodically dredge the navigation channels that lead into ports and marinas. Otherwise, the boats cannot reach them. And the idea is to try to use these plants, because they are sediment traps, they function as sediment traps, to prevent this silting of the channels and to minimize the needs for this dredging, which has a severe impact on the ecosystem and the biodiversity. So we will try to design and validate different placement solutions, the density of the plants, and the substrate that we will use to lock the plants in place, and to try to see if this really is something that we can put the plants also in the use for people. And, well, finally, because most of you probably are not familiar, this is a few photographs of what we've been doing already. The photographs on top are from a harvest campaign that we did in Vigo recently. And the plants that we are talking about, you can see them there. They can have leaves up to 1 meter long, so they really can change the habitat where they are. And on the bottom is the transplants that we already did into the tanks, and you can see them already growing. And this started like 6 months ago, and the plants are for now they are healthy, so it's looking favorable. And because, as I mentioned, this will rely a lot on citizen science and the volunteer efforts, I ask you all, if you are interested, to join us. You can find us online, or you can come see me afterwards, and you can register as a volunteer and participate in these actions. Thank you.
Thank you so much. I think this was very nice to ground us in this place that we are. And we wanted basically to bring us in Aveiro in place. And it's really interesting how you brought seagrass and how nature-based solutions can actually enable us to design. And now we're going to move to Carmen. And please come in. So Carmen will take us now to the laboratory, please. And as you find your presentation, yes? Oh, okay, you can just press the next button. Press the next button. Okay. So you're actively choreographing materials, and from the biodesign, let's say, in nature, we move into the laboratory, so the floor is yours.
Thank you to everybody. And I'm Carmen. I'm a researcher in design at Sapienza University of Rome. And today I will introduce you to the project we are experimenting with a question that is, how can design-driven material and processes experimentation be truly interspecies? In fact, we started from this question because now it's possible to meet different agencies inside the productive context, like biological organisms, advanced machine tools, or disciplines, and they are mediated by the biodigital technologies and possibilities. But how design-driven experimentation can give equal dignity to all these agencies, understanding their interaction dynamics and meeting their different way of knowledge, their different logics, time scales, and so on. So we try to answer to this question, experimenting about bioreceptivity and varying the properties of clay-based materials to make them bioreceptive from different scales. In fact, the project try to play on the meeting of continuities across scales and discontinuities in each scale. So what does it mean? We try to manage the different aspects at the different scales in a systemic and hologrammatic manner, from the intrinsic micromaterial properties to the macro overall geometry. But at each scale, we focused in a biological-like perspective on the discontinuities that are irregularities, porosities, voids, and so on, that, like in nature, can be reevaluated and used like a design mean and not avoided and discarded with expensive energy used. So at each level, we have selected some discontinuities and we tried to make them in a different manner. In the micro scale, we refer to surface ruggedness and intrinsic material porosity that are as discontinuities, and they are given by mixing clay with different waste materials of local researches, like, for example, coffee and cardboard that are very present in Rome territory. We mixed them with clay and with the cooking the ceramics. These wastes are pyrolyzable, non-toxic, obviously, and they create different porosities, and we can manage them changing the quantity and the type of the waste, and so changing the bioreceptivity of the material. At the mesoscales, the discontinuities are the intrinsic variability and defects of the productive manufacturing processes, both digital advanced manufacturing like 3D printing of clay or CNC milling of molds, or, for example, the laser cutting of glazing and so on, and also traditional processes like impression, glazing. Glazing, for example, has a lot of defects that ceramists try to fight, but for bioreceptivity they can be very useful. And at the macro scale, we focus on macro application, so we can vary the morphology and the surface macro geometry of our product to for a specific application. We are now at first stage, and so we have tried to do some experiments, and the first application of this project was a facade for urban regeneration, in particular for the regeneration of the Tiber river banks in Rome. And so we started with these mixtures. We have, firstly, we have varying also the mesoporosity of the surface only manually, but then we started with also other digital processes, like, for example, 3D printing, and also the type and the way in which we grow biological organisms was important. For example, we started from spirulina harvesting, and now we have developed multiple ways to cultivate and biocolonize our surfaces, also with cyanobacteria films that are characteristic of Tiber river banks and moss. We are trying to deep our knowledges to go forward. So the project will finish in 2027, so we have some time, but I'd like to conclude that I want to show you through this project that interspecies design is not a metaphor, but an operational possibility. In fact, focusing on discontinuities, we have tried to understand the way of the behavior of each actant, so the biological organisms, but also, for example, the tool, the digital advanced tool, or the human action to the traditional processes, and try to combine them in order to make a final piece that it's real, produced by everyone. Thank you.
Thank you so much, Carmen, for showing us these continuities and discontinuities with interspecies work, and it is a timely manner. And for that, the connection with the local culture, you know, and how can we grab the biodesign collective knowledge that is in the local culture. So next, we will have Ebru Öner, I believe I pronounced it right, who will show us a very interesting point and also a local point on Polyextremophiles. So tell us more.
Yeah, thank you. First of all, I'd like to thank Nina and Pierre for having us here and introducing our interest. I'm from, so my name is Ebru, Toksoy Öner. I'm from Bioengineering Department of Marmara University from Istanbul, Turkey. And I would like to take you to the world of extremophiles. Actually, when you look to our world, actually most of it is made up of extreme environments. The water, our waters, are actually saline. 97% of all waters is salt water, and only 2.5% of fresh water. If you look to the oceans, 75% of ocean is a very, very high pressure. 75% of the Earth's biosphere is at very, very low temperatures. And the temperatures can go up to 400 degrees centigrade at a certain point. And when we look to the nature that we get all the inspiration for biomaterials research, is actually only a small fraction of the whole picture. So what we humans see as normal is actually only a very, very, very small part. And extremophilia is a matter of perspective. So these environments seems very extreme for us, but there are a lot of microorganisms and plants living in these environments, and for them, this is the normal. And these environments do not come with just one extreme. For instance, if you look to salty environments when there is a lot of salt, but there are tides where the water content changes from season to season, and the soil concentration changes a lot, and there is high UV light, and so all these organisms are exposed to more than one kind of extreme. Radiation, salt, temperature. There are high thermal vents under the ocean, so the temperature is very high, the pressure is high, there is salinity. So we are very interested in such environments. And there are many ways these microorganisms cope with these extreme conditions. And one, for us, very interesting one is that they form biofilms. They produce exopolysaccharides. These are sugar molecules with very distinct properties, and they act as a protective coat, but also they enable environments where these microorganisms, communities, actually different types of microorganisms, they can live and protect themselves. And we believe this polyextremophile exopolysaccharides are actually evolved to survive under multiple extremes, so they have very interesting properties, and these materials can find different uses in as materials in healthcare, food, and biomedical uses. Oops. Sorry, there should be actually the name of actually. Okay, I will. Yes, yes, that's okay. That's okay. Okay, the name of our research group is Industrial Biotechnology and Systems Biology. We are interested in extremophiles and their industry important materials. We worked with thermophiles, holophiles from different parts of the world. And we are not only interested in the production of those materials, but also in their applications in different fields, in food, cosmetics, and biomedical applications. And these are just few examples of the materials that we work on: injectable hydrogels, gels, nanoparticles, microneedles, any different materials. And here was another picture of our, but never mind. Now, currently, we are interested in the genome of all these microorganisms that survive under multiple extreme. We are now mining into their genomes and approach these environments from a systems-based view, what makes these organisms survive under these environments. And if you are interested, we are more than ready to join forces and understand these environments. Thank you.
Thank you very much. So Ebru took us to the limits of where life can exist. And now Emmanouela will bring us back to the human body and to the challenge of building synthetic models for the most, let's say, complex diseases that we face. So I would say that even though the scale shifts, the question remains on how do we work with complexity without simplifying it and dumbing it down, let's say. So the floor is yours.
Thank you very much. Thank you very much, Eleni. I'm very excited to be here with you, and I will be giving you basically a snapshot of our work on human-focused models of pancreatic cancer. Now, why human-focused? Obviously, we're talking about human disease, but also there's been an interest in the community after the FDA announced that it's going to phase out animal testing for drug discovery to also go towards this direction. So it highlighted how the animals are not the most favorable model for predicting how the humans will respond to different therapies. And although in our community we don't really use animals, we do use a lot of animal-derived components, and there's this growing interest to see how we can replace them with either synthetic or human-derived components. So our work focuses specifically on pancreatic cancer, which is a disease with a very dismal survival. This is due to its asymptomatic nature. So this disease is diagnosed at a very advanced stage, at which point the cancer cells have either metastasized to a different organ, and they're also quite resistant to the available therapies. And how they do this, these cells are able to actually take advantage of their microenvironment. So the stromal cells in the ECM, so the extracellular matrix, they stimulate these stromal cells to deposit large amounts of ECM, effectively creating a sort of shield around the cancer that makes any available therapy kind of impermeable. They cannot penetrate through to find the cancer, whether that is an intravenous chemotherapy or even radiotherapy. And we're able to mimic this histology in the lab using synthetic materials. So we use a polyurethane foam, which is a synthetic material with large interconnected pores. And coating it with different ECM components, we can populate it with either cancer cells alone or in combination with stromal cells, so fibroblasts that are activated, and endothelial cells. And what is good about this model is that we can also control the spatial arrangement of these entities. So we can put cancer cells in the middle and surround them with this dense stroma mimicking the histology of pancreatic cancer. And as you can see on the top right, we can see that these two compartments can actually communicate with one another. We can see cancer cells invading the stroma and also stroma cells going into the cancer core. We can also replicate this desmoplastic reaction, so this increased deposition of ECM components by the fibroblast. And we've seen in further publications that when cancer cells are cultured with this stroma around them, they're a lot more resistant to therapy, so chemotherapy and radiotherapy, whereas when they're alone, they're more amenable to this therapy. Now, this model largely relies on animal-derived components. So the biggest one is fetal bovine serum, which is central to our cell culture. So all of our cell lines are cultured using this serum from calves, and also animal-derived protein coating. So we're looking at which different ECM coatings, how they impact cancer cell behavior. And these are derived from animal sources. So we're using laminin, collagen type 1, and fibronectin. And for the purpose of this talk, I'm going to focus on these 2 points that I will be showing you further on. So I'm not going to bore you too much with the details, but replacing FPS is actually a grand challenge in our field. All of the cell lines we use, although they're human-derived, they largely rely on FPS. And in this case, we've tried 2 different approaches. We've tried either synthetic sera, so chemically defined, that we've bought from companies, or human serum derived from volunteers, male volunteers with blood type AB. And basically what we see is that with a synthetic serum, the cells are not really liking it. So over time, regardless of how we introduce this synthetic serum, the cells eventually die. And also, remember, we're doing a multicellular model, so we've not been able to adjust all of our cell lines. We've only managed to adjust stromal cells, so the fibroblasts, but we saw that after reviving them, they also died, and it was not possible to culture them. Conversely, when we used human serum, although the cells went through a period of adjustment, they did manage to actually grow very well for us. They have a lower metabolic activity, and their doubling time has increased, but we were able to get a stable culture throughout, and we can use them in our 3D models as well quite successfully. And finally, we want to replace these full-length animal-derived proteins with synthetic peptides. So peptides are small sequences of these proteins, and basically these are the only sequences that the cells will actually see and interact with to attach to them. So for collagen type 1, we're looking at, for example, GFO-GER, for laminin at ICVAV, and for fibronectin at RGD, which is the most well-known peptide in the community. And in this case, we've tried 2 different approaches, either a free peptide, so the peptide in solution, or the peptide encapsulated in gel in collaboration with Peptimatrix that create peptide hydrogels. And as you can see on the top right panel, we're able, using our own in-house protocols, to coat this scaffold. Remember, it's quite a 3D scaffold with large pores, but still quite difficult for liquids to penetrate through. We've managed to coat this scaffold all the way to the center very successfully when we image with this FITC tag attached to the peptide. And we can also culture cells successfully with even low concentrations of this peptide. So a lot of publications so far have used higher concentrations compared to full-length proteins, but we're able to see that even with the lowest concentration that we use for full-length proteins, the cell viability is actually really good. So this protocol currently relies on physisorption, so weak bonds between our polyurethane and our peptides. But in the future, we want to actually chemically modify the polyurethane to attach these peptides with covalent bonds to make this coating even longer lasting and see what effect it will have on the cancer cells, depending on which peptide we're introducing to it. So hopefully soon we'll be able to bring these two together, so cells growing in human serum with a scaffold that is fully synthetic as well. Thank you very much for listening.
So, thank you so much, Emmanouela, for showing us the beauty and the nightmare of biodesign. So it is our challenge and our job here to think about how to couple the biotechnology to tackle a fantastic human challenge about pancreatic cancer and to cope with replacing what the business side, the corporate side is currently using. So thank you so much. And in an attempt to answer this, I invite Maria to join us to show us how can robots try to assemble human tissues, right? So.
Thank you. So my name is Maria Clara, and I need to thank Emmanouela for introducing a little bit the theme of tissue engineering. That's the topic that I'm going to show you. And we are trying to imagine, like Emmanouela said, trying to mimic a 3D model, a 3D tissue in the lab. Either is a healthy tissue or a disease model is not easy because human body is quite complicated. The architecture is quite defined. So trying to build that in the lab is not easy. That's why we are trying to use a different approach that we call the 3D living tissue, where we try at least to take advantage of robots to help us build a tissue. So, okay. So in tissue engineering, we have two different approaches. The first one that people start using is the top-down approach, where we start from a prefabricated scaffold and we seed cells on top of that scaffold. Although this might work for some application or for some tissues, it has some limitations because we don't allow cells to spread as they want and they cannot control where they want to grow. We cannot control where to grow specific parts of the tissue. To overcome this part, researchers are using now the bottom-up approach that we start from the single building blocks that can be single cells, microtissues, micro units, and using a layer-by-layer approach, think on like 3D bioprinting, that is a bottom-up approach. We construct the tissue that we want and we can spatially place the cells or the microtissue where we want. So taking the bottom-up approach in mind, and also inspired by nature, that is in this case is mussel and the way that mussels can adhere underwater to rocks. We created a capsule, try to think like boba beads in bubble tea. So we were able to take advantage of dopamine, that is a catechol molecule that mussels have and allows its strong adhesion to rocks, and we included dopamine in the shell of the capsules, and in the side we have a liquid environment. And that environment gives everything that cells need because it allows the nutrients to diffuse. The gelatin shell allows cells to adhere, but they are free to move inside the shell to create their own matrix and create its own architecture. And because the shell has dopamine, we are able to create a sticky capsules, and using, as we call a sticky chemistry, that where dopamine oxidized, we were able to assemble these 3D structures that you can see there, starting from a micron unit to a centimeter cube structures. Okay, so because not everything is like as straightforward, we also need to take some design consideration, like for instance, the contact point between the capsules, because as you imagine, if you want to create a centimeter cube structure, the capsules need to be stable enough and the final structure need to be stable. So in this case, we found out that the contact point between one capsule and the neighbor capsules are quite important. So we came up with an offset configuration where one capsule could interact with at least 3 other capsules. We can also, and we also need to take in consideration the size of the building blocks, because if we want to be specific with the architecture, perhaps we could go with small capsules. If we have a tissue that is not that intricate, we can go with bigger capsules. But for instance, if we want to use a capsule that is more or less 1.5 millimeters, that is not small, for instance, for designs, perhaps it's small, but for us it's super big. We need more than 200 capsules to have a cubic centimeter structures. And also the building pattern, because each capsule can have its own environment inside, we can create a structure and different patterns for the final structure. But imagine going to the lab or taking like Legos, and you have 200 pieces and you need to assemble it by yourself. It takes time, and in biology it cannot take time. So we need to do things faster and as precisely as we can. That's why we are using a robotic arm that is like this size, is quite small, and it has all the movements. I don't know if the video will work or not. It has all the movements that our arm has and our hand and so on. So we were able to create in the lab a grip system and an aspiration system. Oh, it has here, yeah. No? No, sorry for the sound, but this is something that we cannot deal in the lab, so at least you have that in consideration. So basically, we took advantage of the robotic arm. We create our own aspiration system. So we created a system that could pick up the capsules and place it in the exact coordinates. And at the end, we were able to create a 3D structures using the bottom-up approach and the layer-by-layer technique. Each capsules have cells inside, and you can see there in the green that are live cells that were able to adhere to the inside the capsule. They can produce its own ECM and can create its own tissue. And because this is gelatin that is part from collagen. Of course, this is animal derived in this case, but we can, cells can degrade gelatin over time with its own enzymes and can replace it. So by the end, if we are able to grow a tissue, we can have something that is 100% made of cells and the ECM of cells. So in this case, we are using the robotic arm to give us spatial control, gives us some reproducibility, mainly minimize human interactions, because in biology sometimes this is a plus, because unfortunately we can bring other stuff to biology, like microorganisms that we don't want in tissue engineering, at least. But we are facing some limitations, so not everything it's heaven. So depending on the living tissues, it might be a problem, because imagine a robotic arm that cannot sense the delicacy of the tissue can destroy the tissue. Also, the specialized programming skills, because biology is, although now it's more common for us to code and to use Python and so R and so on, but imagine a master student that developed all the work, a biotechnology master student. She was able to create the gripping system, the aspiration system, and the code. And the other thing that it's really important is the real-time sensing, because as humans, when we make a mistake and we notice the mistake, we can correct the mistake. Robotic arms cannot. If it makes to pick up a capsule, it doesn't know, so it keeps up the code, and we don't have the final structure that we have. But that it's fixable, because we can add a real sensing to that, and some researchers have been doing that to try to do that. So for now, our system is not fully automated, what for us is not bad because we can learn with it and we can still have a part on research. And I want to acknowledge Ana Margarida because this was a master student that she was able to do all of this part. Although she's a biotechnologist, she took the leap. And I also want to acknowledge DESIGNAE for taking the leap to join designers and biologists, biotechnologists together and try to understand both worlds. Okay, thank you.
Thank you. No. Yes, so now it's time for questions.
We're on time. And we are on time. So might people join here so that everyone can see everyone? We have 3 quick questions, and me and Cristiano probably have some more longer questions, but we'll see if we have time for those. So, okay, everyone fits, please. Okay, so we have for Carmen. Okay. What methods do you employ for assessing bioreceptivity on micro and macro scale between different scaffold iterations? And there is a follow-up, so I'm going to say it right away. Do you aim for a demonstration in the city as the prototypical full-scale application?
Thank you for the question. And before, I forgot to say that Sapere & Co is an interdepartmental center, so it is very interdisciplinary, and we have the help of a lot of knowledges, and it is a fab lab, so it's very experimental, and so we work a lot with learning by doing. As regard methodologies, we at each scale we focus our attention to one discontinuity and try to assess the different variabilities in order to understand the behavior of the other agents. So, for example, in microporosity, we have tried with different kind and percentage of the wastes to understand how the porosity of the material change. Now, at the mesoscale, that is focused on the surface microgeometry and the infill structure of the scaffold. We now are focusing on 3D printing of clay. So, for example, we have selected some discontinuities, like, for example, the layer, so we are trying to vary the height, the nodule diameter, and so on, and try to understand how this variability influence bioreceptivity. At the macro scale, we try to focus on the external condition and environment, so adding some parameters on the project. So, for example, we focus on light, humidity, and other sound and other characteristics of the specific environment, and try to modify the morphology of the structure for add some properties, for example, sound absorbance. So then selecting for each scale one interesting path, for example, mixing with coffee and a specific layer 8 nodule diameter, and then a specific morphology, we try to choreograph the bioreceptivity of the material and obtain a specific pattern, but also aesthetic and performative properties. This for answer to the next question, for the final prototype that is an artwork in collaboration with the Modern Art Museum of Sapienza, that is an interactive installation on the banks of the Tiber River, and it wants to communicate with aesthetics, this interspecies collaboration, but also to be multidisciplinary and multi-experiential for different kind of people. So we will focus on different senses, touch, sight, so on. So there will be a prototype.
Yeah, so that sense of aesthetics interspecies design sounds amazing for us. So the next question is for Ebru. What do we see as the biggest challenge for working with polyextremophiles, especially when thinking of commercialization of possible
products? Maybe.
Yes.
Yeah. Thank you for the question. I think the biggest challenge is to mimic those conditions in the lab, and trying to understand how these microorganisms or these living things survived under those multiple extreme conditions, and how to make these organisms produce these polymers, because we are after those biomaterials and it's not easy to understand them and make them produce these polymers. How to make them attach to surfaces, make biofilms. These are not so easy to understand as humans. So this is the challenge. Thank you.
Thank you. Okay, to Pedro next. So someone is asking, are you planning on germinating the seagrass yourselves, or will the project be strictly transplant-based?
Thank you. Yes, the project involves the development of a seagrass nursery to develop and optimize methods for seed harvest, seed maintenance, and seed germination also. And for the nature-based solution, the idea is to plant the seeds into the structures and then have them grow and then put them into the system.
Okay, thank you. So a quick one to everyone, and then we have some questions for all. So, okay. So Emmanouela, wasn't there a problem with batch-to-batch variation of human serum, they ask?
Not yet. So obviously there's the same issue. FBS comes from an animal, human serum, again from an animal, but from humans. So for now, we're only testing with one batch of human serum. So we still have to order serum by batch, and there is that potential drawback here as well. So the plan is, to probably follow the same principle of FBS, order the same batch of serum. So hopefully a problem will arise on our next order. But eventually we want to explore synthetic serum a bit more. We haven't had the time to do it, so it could be the case that we need to increase the synthetic serum, and in that case we can get past the batch variability. But for now, no problem so far, yeah.
For now. Keep in mind. So the next question is to Maria Clara. Which tissue types do you think this approach can be suitable for?
So I think we can, depending or making some chemistries, we can target any tissue or model disease, because in this specific case, I'm targeting bone, because we found out that the catechol can also improve mineralization. But if we play with the chemistries and other trying to mimic the real tissue or the environment, we can use either tissue that we want that needs a specific architecture and it makes sense. And another advantage is because this is a liquid environment inside, the nutrients and oxygen diffusion is not a problem if we want to go big. It might be once cells start to producing ECM, in one point we need to include vascularization, but it's something that we can find in vivo as well. So I think we can try to target any tissue, meaning like trying to see what kind of environment there is there, and we're trying to shift for the lab.
Thank you. So now a question for all, and maybe each of you can give us a bit of a short answer so we are in time. What is your perspective regarding the collaboration of your field with design regarding the objectives of this work group, which I will remind is a bit of creating a common language, finding terminology that we can understand, both designers and scientists like you. So if you can go around and tell us a bit, that would be nice. Yes, that's a difficult question.
Yeah, I can start. I think the mindset is a little bit, it might be a little different, but at the end I think we can speak to each other. I took a venture and took 1 year on user experience and user interface, and I'm from biology, so it was not easy to see user-centered, but now here we are trying to shift for human-centered, and I think from my perspective, and I joined it after the cost action was formed, and I think it's really nice to try to make us understand each other and trying to and it's perhaps an easier way to show other people, the common public, because I think it's easier for them to understand more the design parts because it's more visual than for me being here and say, oh, I'm creating a tissue in the lab. Yeah, okay, a tissue. How? People are imagining a lung. No, we don't do a lung or a heart. Like we do a tiny thing that is not beautiful, it's ugly, but it mimics because we don't build human bodies yet. And I think this consortium will help to shift that mindset for the other public to understand a little bit what we are doing in our part. I don't know, and I would like to know how designers see ourselves, because here we are more scientists. I don't know how it's the other way. I would like to know, and what was the main idea when it came up to join designers and scientists, because I'm on the other part and I found out from the scientist part. So I think it could be a good synergy and a different way and good way to showcase people what we are doing. And sometimes I talk for myself, it's not easy to communicate scientific words here. I try my best today for you to understand a little bit what we are doing, and I think designers think more about how to communicate and they are more creative at the end. Sorry, we also are as scientists, but I think the mindset that designers have are something that we need to take to our field and to think outside the box and see things differently.
Yeah. I can add to that. I believe every project that was brought here today has a multidisciplinary perspective, and I believe the different views into the same problem are always the best way to have a look at the problem, because as scientists we think about the solution by a determinate way, but then you can have a completely different mindset and a completely different view that can many times resolve a problem that we cannot. And I think this balance between different fields of work is it can be very helpful for us, and hopefully we can also help you in your work. Thank you.
So I think I am the only designer here, okay? No, no, don't worry. So maybe my perspective, I agree with you, but I think that the best way to collaborate and communicate is by doing. So we can establish univocal aims and also at the same time understand us very well, because scientists have vertical knowledge and also interest to go deeply on questions. You know? So they can focus and arrive to the final result very well by deepening the knowledge. Designers maybe are more horizontal, so they don't have the abilities and tools to arrive to these final results, so they can be helped by scientists in this, but they can give a broader perspective and, for example, a final application in society to the researches. So by doing together things, maybe there is more interaction and also more exchange of what can be useful, the scientific research for the society, but on the contrary, what things we can deeper and understand well together to another for other opportunities and so on. So, thank you.
Yes, I think Maria covered a lot of the tissue engineering side. So the only thing I would like to add is actually, yeah, we are inspired by nature, but replicating this in the lab can be a bit more tricky to do. So I'm a biologist by background, so going into tissue engineering, I had to think a lot about how do I design something new in the lab. So my PhD included a lot of thinking, computer-aided design, and doing that from a biology background is a bit tricky. So having other people more experienced in this field, I think, is definitely very, very valuable to actually come up with ideas. So even for like bioreactors, for example, where do we house all these scaffolds, all these tissues? There's a lot more to think about there. How do we design a bioreactor? How do we make it work? Bringing engineers in, I think, is definitely important. So the interdisciplinary nature of our work is, I think, central. And there's so much that each person can do, but when we come together, I think we can achieve a lot more, definitely.
Thank you. I agree with all the comments. I just want to add my perspective, what I expect from this action. In our research, we want to translate the products of this extreme world into some products that we can use under extreme conditions, physiological extremes, for instance, deep burns, deep cuts, which products that can work under those extreme conditions, but not just tissue engineering, not just physiological extremes, buildings that can resist extreme environmental conditions. So this is our aim, and in order to do this, we will need some perspectives from different backgrounds, textiles, extreme, that can resist extreme conditions, and I'm very happy to be part of this. So,
thank you all. I think we are on time, and do you have anything else to say? We have more questions? No, I think we're good. Thank you all for your contributions, and let's continue.
Yes. Thank you very much. Thank you, Eleni. Thank you, Christian, for the moderation of the session. I think this went really, really well. I'm going to invite you now for the coffee break, which is outside. The bathroom is right on this side, and I will meet you again in 20 minutes.
Welcome back, everyone. I hope you had a good break. I want to give the stage now to Victoria and Ruben for work group 2.
Thank you. Yeah, welcome. This is work group 2, which is focused on biomaterials, biological mechanisms, and fabrication. Just a quick introduction to who the leaders of the work group are. So I'm Victoria Outram from the University of Strathclyde in Scotland. I'm a chemical engineer. You have Ruben, who is a marine biologist working at ECOMAR here, and Eliza, who is in the middle there, who is an architect at the University of Stuttgart. So in this work group, we focus on 3 main aspects associated with biomaterials, and that's scaling and translating bio-inspired structures and processes, assessing and improving the sustainability of biogenic materials, and also looking at, we're also looking at developing scientific and legal guidelines for the biodesign implementation. Within this work group, we're very multidisciplinary. We probably cover all topics of people here today. And we've kind of got, you can see this balance within our disciplines as work group leaders. So this session's split into 2 panels, and we'll start with our first panel, which is on biomaterials, biofabrication technologies.
So, and to start, we will start with Blarina, that will tell us more about the biomaterials in textiles.
Thank you very much. It's black.
I don't see the screen. Okay, so thank you very much for introducing me. Thank you also, Nina, for inviting. It's interesting project. My name is Blerina. I'm coming from Polytechnic University of Tirana. And today in my presentation, I'm going to talk about the textiles and the importance that they have. Well, we all know actually what are textiles and where we find them. Even in this room, without taking into consideration the clothes that we have a lot of textile elements. So they are everywhere actually, and we can see them. And of course, they started quite early actually as a need for us. But the thing is, the situations recently actually, not probably very recently, but quite long, maybe 10 or 20 years ago, it started to be worse because the textiles that we were using were producing a lot of waste. And a part of considering, in the past actually we didn't have such kind of problem or troubles, let's say, because the products were biodegraded. One thing very important. And the second thing, at that time we were not using a lot of textiles, so we are not overproducing textiles as we are doing right now. Because people are really focused on the performance, they really want to change their clothes every day. And since they wanted to go in this direction, it means that the industry needed to answer to their needs. So somehow we have to blame ourselves because we are in needs all the time. But on the other hand, somehow we have also to blame the industry that they are overproducing. So, and also, since we want everything to be perfect, we started to replace natural fibers with synthetic ones, which means that they are a real problem because they are not easy to be degraded. So it takes hundreds of years to completely vanish. And that's why we are in this situation having 7 million tons of textiles, and actually that's only in Europe. Imagine what will happen on other countries that they are not reporting exactly what's going on with their textiles. So it means that the values that you see there are even higher. Actually, when we talk about waste, it's a thing that we see in this graph. So the thing that we are measuring easily. But how about to talk or thinking about the line of production of textiles, starting from fibers and going to yarn, fabric, and finished textiles, finished fabric. So in each step, jumping from one step to another one, let's ask ourselves, what is the energy that it has been used? How efficient have been all of these steps? So how efficient the products were or the product was. So from spinning, what is the amount of the energy? What is the amount of waste? Or to the knitting, what is the amount of water that needed for these textiles? So how efficient are we in all of these steps? Also considering the dyeing and finishing stuff. Are we taking natural dyes or are we going for synthetic ones? And how can we replace? So there are several questions actually that really everyone actually can ask and think about what can we do to change a bit the situation. And of course there are people who are thinking for solutions, and I brought actually several examples here. I just chosen randomly and without any specific point, just to say that actually people are thinking to replace these classical textiles with other products, based, of course, on natural elements, as it is tencel, which is completely based on orange, and deserto. They imitated the leather, but by using the nature. Algagnite, spiber, and design to fade. So all of these actually are examples to bring here and to say that the solutions are there, but what we have to change is the technology, probably, how to produce these textiles. Should we skip with the classical way of producing, or shall we change to another, to jump or to shift to another way of producing? To think about linear producing industry, or to think about a circular one. And actually, these are the main challenges. Scalability seems to be another problem. Of course, these are the solutions, and most of them are coming from lab, but not going directly to the to produce for the whole population. So scalability seems to be a real problem. And also, what about the cost? Are these textiles affordable for everyone? Performance and consistency. So we are used to have textiles and to perform in the best way. So it means performing on the when we are wearing them, but also to resist different kind of mechanical behaviors. So are we able to produce these textiles and to answer the new or the performance that we used to test before? Circularity seems to be a good thing here. I mean, for these examples that we are bringing here, and for other examples that they do exist. But are we sure that they didn't add other synthetic materials that are not possible to vanish during the time after the use? So navigating actually all of these challenges will give us a trajectory of the biomaterials as a revolution that is coming. Normally, by bringing these problems and challenges, what we can say is that biotextiles actually are a must if we are still thinking about nature. And of course, are essential for transitioning the textile industries towards sustainability and higher performance, functionality, reducing reliance on fossil-based materials, and reducing carbon emissions, which seems to be a real problem right now. The future of industry lies on the concept of programmable biology, where cells are engineered and programmed as programmable factories to solve specific problems, from creating high-performance fibers to producing non-toxic dyes. Actually, that was a kind of conclusion that I wanted to add by bringing several problems that the textiles are facing and are introducing as a pollution to all of the globe. Because we have pollution on water from textiles, from synthetic fibers mostly. We have pollution on the salt and also on the air. Recently, I read an article where it was mentioned like from the air are raining microfibers. Imagine what's going on actually on the nature. So considering all of this, actually we have to think how to solve the problem. Considering the fact that textiles are everywhere, so we really have to consider how to find a solution for replacing the textiles and to come also to the sustainability. That is the thing that everybody is thinking about recently. So thank you for your attention.
Thank you very much. That was a nice presentation. Yeah. Next up, we have Sem, who will be talking about controlled biomineralization and self-cleaning polymers for nanofiltration membranes.
Thank you. First of all, Thank you for inviting me to this working group and action. I am Cem from Hacettepe University, Ankara. And today I want to share our findings and our research in how we, inspired by nature through modification of metallic implants. Some of you may know, or some of you may not know, currently in the last years, we are not using passive scaffolds or passive implant surfaces for the tissue engineering or biomaterial science. Instead of, we are using some active ones in the organic material mainly, because through this chemistry language, through some chemical functional groups on these kind of materials, cells and tissue can communicate with each other through the language of chemistry. But in the case of metallic implants, especially titanium, which is the gold standard in orthopedic devices, as you may know, there is a lack of communication between the organic milieu and the metallic ones, because they are lack of, let's say, alphabet of organic groups, so they cannot communicate with each other. And through this emptiness of lack of communications, cells find themselves in a very awkward and lonely in the top of the surfaces, because they are not communicate with the surface, and the metallic implants, the only communications that they can interact is the topography, which is bone itself. In the natural ones, the bone itself also has a nanotopographical, hierarchical organization. There are osteons, Haversian canals, nanocollagen fibers, nanohydroxyapatites. They are periodically well aligned and making themselves in a good hierarchical structure. And through the years, we are trying to mimic that type of topography, and or more sophistically, we are trying to give titanium surfaces a voice that they can interact with the especially stem cells and the osteoblasts that can communicate to each other. This is our setup is called anaerobic oxidation. I won't give the details in this chapter, but if there are anyone interested of it, I can give some more detailed information in the lunch or coffee breaks. Why we choose this kind of nano tubes or hexagonal array structures? Because it's not just their fancy-looking nano-hierarchical structures in nanoscale, but also through the nature's evolutionary timeline, the hexagonal structures or honeycomb conjecture is exhibiting a maximum internal porosity volume that they can interact with the rest of the material that they are in junction with, or they have a perfect mechanical load distribution, isotropic distribution, when some materials, especially cells, that can spread on this type of level. So with this approach, we are trying to make bioinstructive structures on the metal surfaces. We are working with medical genetics department of our university, and in one case we tried to see what was the conclusion of this dialogue between the titanium surfaces, bioinstructive surfaces, with stem cells. So what that surface instruct to what does that surfaces instruct to stem cells? And we saw that mainly there are some genes upregulated more and more through the especially extracellular matrix protein synthesizing genes. And unfortunately, not unfortunately, we try to variate some kind of surfaces, but with the same surfaces, same materials and same cells does not generate always the same results. And we saw that while they are communicating with the surface, since the geometry is the same, but if you change the diameter of these hexagons, nanostructures, or the thickness of the walls, in one condition, since they are the same chemistry, the cells are very happy and spread this as much as they can. And in the another one, the cells cannot spread, and always in the mobile phase, seeking for new energy or new place to go where they can make themselves much happier. And through this search, some of them are going to die in this exhausting search. And also there are some genes that are related with the mobility and vascularization. They are much more upregulated than instead of making extracellular matrix, which we called their home. So if we make an useful and explanatory analogy, what we are doing, we are trying to redesign the implant surfaces with an inspiration by nature. So we can also make some analogies. Let's say there is a climber climbing on the mountain, and in this scenario, that bounce holes are maybe RGD sequences on the surface. Handhelds are integrants, ropes are focal adhesion complex, and the mountain is the implant surfaces. We're trying to redesign that surfaces that the climber making themselves much more comfortable and enjoy the landscape instead of getting stressed. So we are trying to make the surfaces much more suitable for the stem cells. I mentioned about there is a nano architecture, hierarchical architecture in the bone tissue. And another thing that we are planning to do is we are currently trying to grow biominerals at the bottom of that hexagonal nanotube structures, because we know that there is tiny little hydroxyapatite minerals that can govern the mesenchymal stem cells and the osteogenic factors in this tissue. The biggest problem is we are still trying to overcome the surface tension of the aqueous media, and the capillary forces works for us to seed that kind of biomineral seeds in the bottom of that hexagonal nanotubes. So as a sum up, we are trying to redesign the implant surfaces because the implant surfaces, we want them to be active in biosignaling and the biomineralization at nano level, instead of just passive nature, just a fixation device, rather than currently we use in the current practice. We are trying to making that surfaces like the action manifest says, from a techno-centered design to a nano-centered one, inspired by the nature. Thanks once again for inviting me, and thank you.
Thank you, Sam. Like a really interesting work, just showcasing how far we reach, even going into the medical field. And now we will have Jedrius, I hope I'm pronouncing it right, coming all the way from Bulgaria to talk to us about self-cleaning PVDF MOF nanofiltration membranes for micro and nanoplastic removal from wastewater.
Yep. Hello to everyone. Nice to see you, and I would like to present one of the topics what we are doing in our department. And actually, I will propose the idea how to solve the problem which was presented in the first presentation, how to clean the water from micro particles. Okay, so maybe it's idea, and maybe we can discuss about the collaboration. And yeah, so textile or actually human is a source of pollution, and with different kind of micro nanoplastics, heavy metals, carbon, and so on. So it's really a huge problem when we talk about the pollution, and we try, actually in our department, we're trying to deal with three problems, and one of them is micro nanoplastics. And we're trying to fabricate membranes. Membranes itself, there is nothing new. Yeah, we can find a lot of information about membrane fabrication using different techniques, and this is just one of examples what we did. Membrane produced from polysulfone with which was doped with chromium MOF. What was the idea exactly here? What we did and what we wanted to achieve, that microplastic itself has charge, negative, positive, yep, and we integrated chromium MOF in this case in order to attract micro nanoparticles with specific charge in order to increase the filtration. And these particles you can see in the SEM images, they are visible. And we're trying, of course, different concentrations, different charge, and so on. And this, how to say, this performance usage of this MOF particles, yeah, it's good itself, but and it increases the filtration efficiency. However, membrane is plastic again, and it's the second source of waste, of pollution, and so on. And idea, what we propose in the next step, is to extend the lifetime of these membranes. Because during the filtration, yeah, it's not a secret, the pores of membranes will be clogged. And due to this, yep, we need to replace membranes. We need yeah, to change, to maybe to change the properties, thickness, and so on and so on. Our idea is to increase the lifetime of these membranes using ultrasonic cleaning, to combine, yep, to use active methods in order to clean the surface of the membrane and to extend their performance. Again, we have two ideas. One of the ideas is presented here, is using some ultrasonic waves, we can clean the pores of these membranes. Yes, it needs some additional design in order to to change the circulation of the wastewater and so on. But the one idea is to use ultrasonic vibrations because our department is strong in the analysis and investigations using different kind of vibrations, starting from micro energy harvesting, using, for example, in milling tools and so on, and continued with different wearable devices and so on. So therefore, we started to analyze this idea to use ultrasonic vibrations for the cleaning of surface of these membranes. Another idea, it's not presented here, but these membranes often are produced from PVDF. PVDF itself also is piezoelectric material, and also we have quite big experience with the production of different films, membranes from PVDF, and idea is to apply the electrical potential to the membrane itself, and due to its piezoelectric properties, we can generate mechanical deformations in the membranes. And this also could help to clean the especially internal pores of the membrane. Yeah, because ultrasonic vibration is good. We know that ultrasonic vibrations can clean the surface, but internal pores, yeah, still could be blocked by filtered micro nano particles. So the idea is to generate vibrations inside of these pores, and in this case, to using the inverse flow of water to clean the internal pores of the membranes. So this is the short idea of what we're doing, and you're welcome to visit Lithuania and to discuss about the collaboration.
Cool, thank you very much. If we can ask all three speakers to sit at the front, and we'll ask some questions associated to this first panel. Is that one working? This one, yeah.
Okay.
Okay, so yeah, as you can see, there's a wide range of work going on within this work group, which hopefully is clear by these three very different presentations that we've had to start with. So our first question for Blarina is, if corporates have all the requirements, so the money and the knowledge around the textile challenges, how can we incentivize them to, I guess, have the power of will? So how can we incentivize them to help solve some of the challenges that you described?
Well, what I tried to bring to my presentation was to work on three different directions. The first one, to change the way how to produce textiles. The second, to change the material. And the third one is to change the mindset of people. Because people want performance, they want to be the best, they want the best textiles. But by two other options that I brought before, so it's definitely we have to change the way how we will perceive the textile. So it doesn't matter if it has a problem, it doesn't matter if it has a defect, or so we have to also check how to test and how to admit these textiles. Mm-hmm. So we have to hardly work actually on these three different directions. Mm-hmm. So it is possible.
And are the big, I guess, textile manufacturers willing to support a change towards, I guess, bio-based textiles?
Well, in general, new things are not easily accepted. Yeah. That one we know for sure. But thinking about the pollution that the textiles cause, I think we have to skip. Mm-hmm.
So now we would have a question for Sam. So Sam, we wanted to know, did you find different transcript gene expressions up or down regulated between experiments? And if so, can you discuss further about it?
Yeah, sure. First of all, there is obvious that there are much more difference in the type of upregulating and downregulating genes, because in the past maybe 20 to 30 years, especially in the bone implants, there is a thing that the porosity and the roughness is good for the implants, but what kind of roughness we can ask first, because they are both in the same hexagonal structure, but the type of roughness is the second one was consisting of very sharp needle-like wall thicknesses, and there are too much empty spaces for cells to spread. The biggest difference in the upregulating factors that if a cell can sit and spread and making itself happier than on that surfaces, they immediately start to synthesize extracellular matrix. We can say that their environment, their home. But if the roughness is consisting of really rough entities like spikes, the mobility genes like CXCR4 or VEGF, which is trying to make induced angiogenesis, that making the blood veins, making blood veins. That kind of energy-seeking and mobility genes are upregulated, and in the other ones, where you can control the nanotopography and roughness, the settling down and the osteogenesis, the bone formation genes are upregulated more.
Yeah, thank you. And the last one, which was done during Jedra's presentation, but I think all of you can answer it. It's basically considering the goal of work group 2, which, might I remind you, it's basically to create a catalog of biomaterials and biofabrication techniques. How could we translate this nanoscale that you guys work on to effectively meso and macro biodesign applications and for the designers to apply it?
Okay, really interesting question and complicated at the same time. And yeah, so ideas always could be applied in different scales. And let's say one of the ideas, so that way, how to say this, what we discussed already, where it could be applied. So it could be, for example, we can integrate natural fibers from textile, maybe gathered or just produced just from flax, hemp, or other plants produce fibers. From them, we can create the biggest scale filters. What can, for example, to increase, to improve the quality of air where we live, what we breathe, and so on. So it's the mixing of different techniques and our competences. I'm sure that we can apply in this work group too, and we can work on development of different materials, databases, and so on.
Well, I can give an evasive answer, like there is plenty of room at the bottom in the nanoscale, but the nanoscience is still in its early stages, I think, because like Feynman said in the 59, we have currently building that tools that we can understand what's going on the nanoscale. So to bridge up this design and the nanoscale materials, I think first of all we have to understand for my view of perspective to get a little bit understand the design strategies, because sometimes we in the positive sciences, in the bioengineering or nanoengineering, sometimes we called our things like we design that kind of material, we design that kind of surfaces, but usually we are not understand that kind of two sides, both the designers and the scientists, I mean positive scientists. I think we must do extra effort to understand our cross disciplines in the first place.
Well, of course, I agree with colleagues. Well, in the middle of 1950s, people managed to produce synthetic fibers by extruding them. So we can just copy the same idea, but instead of using synthetic materials, you can just use natural or biodegradable materials mixed together in order to stand, in order to be able to produce the textiles, and textiles themselves in order to stand. So that is possible. And then going deep into the dimensions, if it's going to be in a millimeter, centimeter, or nanometer, or micrometer. So it's something that still can be solved. So I think it is possible. So.
Good. We have another question about membranes again. Are there any migration or degradation challenges regarding the membranes that you talked about? And can you tell us a bit about the efficiency of the membranes?
Yeah, so efficiency, how to say, itself, it's, as I said, it's not a huge problem. We can achieve really good efficiency, 90% and more. Yes, the biggest problem is degradation and the lifetime of these membranes. So this is the hugest problem, because how often we must change these membranes in the real devices. This waste, also the wastewater also has different conditions, because when we wash clothes, we use different chemicals. Again, it's another problem. Yes, so and for water and for the membranes, it will degrade and so on. So it's really a lot of questions what are not answered yet.
Well, I guess that gives us lots of work to focus on. So one last question before wrapping up this panel. Given that we've got a wide range of people here to network with, is there any sort of particular collaborations that you're looking to develop during this conference?
Well, that's for sure. We have a lot of possibilities, probably with the chemists, that they know exactly how to combine materials together. And we, as a textile engineer, probably we can just think about how to construct the textiles. So that's the very first collaboration that I see actually in this. And then, of course, thinking about degradation, how biodegradation, and also we have to work on how to test these materials. That's another step actually that needs to be solved. And if we have to think about the standards. So are we going to test these materials with the same standards or with the existing standards, or we are going to change? So as I said also in the beginning, the mindset of people is really very important, and that thing can change only if they see the results. And the results probably are on our hands.
Okay, so about collaboration, so I'm from mechanical engineering department, and we always try to think about the application of different results, about how these results, techniques, whatever, how they could be analyzed and applied. And we're open for any discussion about different techniques and application. We're strong in simulations also. We collaborate with the SALT system, and I'm sure that we can create some useful techniques and devices inspired by biodesign.
Thank you.
And also, as a major in chemistry of our site, we always used both. We synthesize our own materials, but always we used the commercial ones, Merck, Sigma, lots of things. And I see that there is a lot of people using that huge biorefinery as a raw material. So we are happy to use them in the biofabrication techniques that we develop in our laboratories through the years. And we are always open for good collaborations, especially characterize them and making them that kind of materials as a starting point to build up new structures, nanosurfaces, nanomaterials, or biomaterials, etc.
That's great. Can we thank again our three speakers for this first panel? So we'll move on to our second sub-panel in this topic, which is focused on biological models, ethics, and experimental frameworks. We have the QR code, the same QR code, so if you can send through any questions whilst these are talking, that would be great. So our first speaker is talking about 3D printing of bio-inspired structures using powder from marine organisms, and this is by Joanna Braga.
Hello everyone, my name is Joanna, and the work I'm presenting today comes from my master thesis in product design and engineering here at the University of Aveiro. The work I'm presenting today, it's called 3D printing of bio-inspired structures using powder from marine organisms. And I hope to show you how design, material science, and biology can work together to make the food system a little more sustainable and a lot more interesting. So I want to start with a number, 21 to 37%. That's the share of global greenhouse gases emissions produced from our food system. And nearly 2/3 of that are linked specifically to animal protein production. Meanwhile, the growing population is intensifying the pressure on conventional agriculture. So we need urgently new alternatives. And what's our proposed solution? Using marine gammarine amphipods, MGA, common known as sand fleas, as the a protein solution. They are nutritionally remarkable, 60% of protein. They are rich in omega-3 fatty acids, and they have a high food conversion rate. They can be farmed in brackish or marine water, and no arable land is required. So their environmental impact is minimal. But we have a problem. They look like insects, and So in Western cultures, that's a significant barrier. So here's the challenge. Having nutrition low-impact protein source is not enough if people won't eat it. Acceptance shaped by the appearance, smell, cultural familiarity is as important as nutrition. And that's where our research begins. And here's where diatoms come in. Diatoms are unicellular planktonic microalgae, one of the most abundant organisms in aquatic systems, responsible for 20 to 25% of global oxygen production and central to carbon sequestration. Their silica shells are the most symmetric, intricate geometries that you'll ever see. So drawing on these forms does two things at once. It communicates a genuine connection between marine ecosystems and sustainability principles, and it redirects the attention away from the origin of the ingredient, shifting the focus from what is this made of to how beautiful is this structure. But how can we fabricate these structures? By 3D printing. It gives us precise control over the mechanical, nutritional, and sensory properties, and that is something that conventional food processing simply cannot offer. So the methodology was inherently iterative for interconnected stages that continuously informed each other. It starts with inspiration and design. So the atom morphologies were studied and converted into 3D printable models. In parallel, we worked on formulating the food paste after establishing a liquid-solid ratio of 55 to 45%. Then 3 functional ingredients were added: green banana, carob, and coconut in different concentrations, 10, 20, and 30%, both individually and in combination, alongside fixed levels of xanthan gum, sunflower oil, and water. So we grinded the MGAs and we mixed with the other ingredients, loaded the printing cartridges, and then the paste meets the 3D printing process. And the printing parameters are continuously adjusted, like the extrusion pressure and the nozzle speed. And finally, the printed structures went through post-processing, either air drying or freeze drying, which had a significant impact on the final outcome, with the freeze-dried samples showing a higher structural fidelity. Here you can see all the final printed structures from 11 different morphologies and designs. And to sum up, we used form as a strategy to turn unfamiliar proteins into something meaningful and appealing. Within the frame of regenerative biodesign, this kind of work, although still early stage and still full of open questions, points toward a design approach that doesn't just respond to sustainability problems, but actively participates in shaping the food culture. Thank you so much.
Thank you, Joanna, and a really interesting work that it's way too close to home. I really like these thematics. But now from something that I really like to something I don't really understand, we will have Maria coming all the way from Greece to talk to us about biofabrication of a 3D breast cancer model using biomaterials to assess the effects of anti-metastatic drugs.
Very much. Thank you for a kind introduction. And hello, everyone. It's a great pleasure to be here in Aveiro. Thank you to Nina and all collaborators and people who participated to this great, wonderful organization of this nice first conference of DESIGNAE. And I'm happy here today to present some of the recent data in my lab. And this is a work that I'm going to show from Ioana Zorba and Varvara Platania, a PhD candidate and a postdoc. In my group, we work for many years on biomimetic, naturally derived, but also synthetic materials, biomaterials. We focus mainly on the biocompatibility of the materials when they interact with cells. And in the last 7 years, we have invested a lot in the direction of biomaterials processing, which also is part of it is biofabrication in order to fabricate three-dimensional biomimetic constructs which can be useful either for regenerative medical purposes or as models to study, for example, to screen drugs. This is the topic, you know, already you have seen the biofabrication of a breast cancer model. This is one of the models we work on, particularly for highly metastatic, very highly aggressive breast cancers. And we used in this case as well biomimetic materials, and we assessed, today I will show only one drug that we have tested, and the role of this drug on the cell proliferation and the cell migration, mimicking the metastasis of the cancer. So this is a brief overview of bioprinting, and you have heard already this morning about printing, 3D printing, and using bottom-up, top-down processes, which are very, very important, different approaches, but they mostly lead to the same aim, to regenerate lost or defected tissue or even an organ, even if it is at a lower level, because organs are highly complex and it's not easy to recapitulate them 100%. They are also multicellular systems, not only monocellular as we usually study in vitro in our cell culture laboratories. So this is, as I said, the overview. So mostly I would like to focus on, this is a bioprinter, an extrusion bioprinter. I try to put the cursor, I'm not sure if it is visible. We use a bioink. A bioink is an ink, a biomaterial, which is mixed with encapsulated living cells. So the cells need to be living during the process and afterwards, during the processing. So this is the bioprinted structure that we can get, and the bioink, as I said, contains the living cells inside the matrix, inside this polymer network, and this is the product of our model. So my student Ioana, I wanted to design a bicompartmental model, which is shown here, bicompartmental because one compartment to the right, here to my right, shows the breast cancer, which is typically of higher stiffness. It is a tissue with mechanical properties like higher values of Young modulus, elastic modulus, but also biochemically has a higher content in collagen. To the contrast, to the left here, we can recognize there's somehow the part that mimics the physiological tissue, which is of lower collagen content and lower stiffness. So two characteristics, the composition regarding collagen content, and the second one is a biomechanical characteristic, which is the stiffness of this. And we could do that by using different combinations of biomaterials. And this was another optimization, how to create these inks in order to receive a higher content, lower content of collagen. In this case, we use gelatin and combine it with alginate, and also the mechanical properties of this one of these two compartments, as you can show here in this model. So a less dense to the left and the higher density, stiffer part to the right. So this is the main motivation behind high density of cells encapsulated within this model, usually 10 million cells per ml. So we studied this stiffer next to the close in the proximity of a stiffer compartment, so like two phases, one next to the other, as an adjacent physiological tissue analog close to the cancerous analog. And this is the model here. Collagen is one of the parameters, as I said, stiffness is the other one. And the drug that we used here to test, this is a ROCK inhibitor. So this is the name of the molecule, Rho-associated protein kinase inhibitor, and this is the commercial name, which inhibits cell migration, but also invasion and cell proliferation of these breast cancer cells that we applied in this case, and they were the 4T1 highly metastatic breast cancer cells. Here you can recognize in this graph the one is in green. We can see the less stiffer, and in red is the stiffer matrix, and we were able to define the Young modulus values, which are, you can see here the values are approximately close to 50 and close to 90 kilopascal, and these are very close to the values that we read in the literature, which are approximately around 30 and 90 kilopascal for each of the tissues, the cancerous and the adjacent physiological one. And to the right, on day 1 and on day 5, in green, you can recognize the living cells stained by calcium life dead assay, and a few dead cells, maybe I can see also here in red. Of the two matrices, again, the less stiffer and the stiffer matrix here designated also with the percentage of each component. In the physiological matrix, we incorporated fibroblasts, while in the cancerous-like matrix, we incorporated the 41 highly metastatic breast cancer cells. And here, in looking into cell migration, we could see these are 4 images, but they are based also on videos, very short videos. It's a time-lapse microscopy that we got from this sample here, hydrogels and cell culture medium in direct contact with the bioink that we have created. And in this case, we pre-incubated with the drug for 2 hours. In the second case, we pre-incubated with the drug for 24 hours. And if these videos would play for a few seconds, we could recognize how does it go in this period of 24 hours. Thank you very much, Nina. Thank you. It's possible. Can you? It's not a problem, but it's not inside. Okay. Because doesn't matter. It's no problem, but if you're interested, I'm very happy I have them with me in my computer, and I'm happy to show how the cells close the gap, because this is a so-called, this is a test for cell migration, but what we measure is how this gap closes, how fast. So practically, for those of you who are familiar with this, like a scratch assay, practically it is a scratch assay, and we see how this gap is closed over time under the different conditions. In the control condition versus pre-incubation with the ROCK inhibitor, which is the drug that should avoid, or it practically inhibits cell proliferation and cell migration. We have quantified also this data, and this was, as I said, in two-dimensional system, because we added pieces of the bioink on top of this cell culture. Looking into a three-dimensional situation, which is much more complicated, and visualization using microscopy methods is not an easy task, let's say. It's quite challenging to follow the movement of cells in 3 dimensions. Even if we use high-resolution confocal images, we do also. It's not always very easy to follow this in a 3-dimensional structure. So here you can see day 0, 1, 2, 3, and 6, and you may recognize a few cells that leave this dense matrix. The dense one is to the left. This is the control, and this is the one with the drug. So to the right is the dense cancer-like, and to the left is the physiological matrix, and we are able to visualize and to image a few cells that leave the stiff matrix, the cancerous part, and they migrate towards the physiological part. So we did many of these assays using fluorescently labeled cells, the same cells with fluorescence, 41 GFP positive cells, and we have a lot of data now to show, and they really prove that there is a difference in the metastatic potential, but also in the proliferative capacity of the cells in the presence of the drug compared to the control without drug. Sorry, I kept the 5 slides, so I have no acknowledgements, although I should. So thank you very much for your kind attention, and thank you again for the organization. Happy to discuss.
Thank you very much. Yeah, we'll invite you back for questions after the next speaker. So we have one more speaker in this section, Eva, who is speaking evaluating biodegradable frameworks or metal-based frameworks for bio-based materials.
Hello, thank you very much for invitation. My name is Eva Jablonska. I'm from University of Chemistry and Technology in Prague, in Czech Republic. And I'm biochemist. And we are dealing mainly with degradable implants for orthopedic applications. We've already heard today about titanium implants, which are intended for permanent applications. They replace the tissue in the body, but in our case we are using the degradable implants, which are intended for example, complicated fractures, and they should, as you can see at the picture, degrade as the bone heals, preferably in the optimal rate. And we are mainly dealing with magnesium-based and zinc-based materials. I'm not preparing them. They are prepared by my colleagues within the university or at the other institutes in Czech Republic, and we are testing them. Recently, we've tested the material which was zinc-based and had a biomimetic layer on the surface from parasholcite, which is calcium phosphate compound and should mimic the bone structure and composition. And these materials, there are many of them, and the main issue is the degradation rate, the corrosion rate, and the biocompatibility, which can be somehow optimized by the composition of the material, and also by the fabrication process, and also by these layers, for example. I'm doing the first part of the testing, which is in vitro testing using cells. and then the materials can go to in vivo tests and then to clinical trials and hopefully then to the praxis. The first test which is required is according to the standard where the material is submerged into the medium, cultivation medium for the cells. The cells are seeded to the wells, and then the second day the extract of the material is added to the cells, and then the toxicity is evaluated because when the cells are alive, they are metabolically active and they can change for example, the color of the substance which is added, and then we can see how the cells were viable or not. But the question is whether this test can simulate the real situation in vivo in the body. And of course not, it's very simplified test. And therefore we are doing also other tests, like we are growing the cells on the materials, on the surface of the materials and so on. And we are also trying, sorry, there's no animation. We are also trying to use the appropriate cell model, and we are also playing with the media which we are using for the extraction. For the material testing, the cancer cells are normally used because they are easy to work with and cheap to work with, but they not always reflect the situation in the body. So we are trying to use the cells which are not cancer cells. They are only immortalized, conditionally immortalized, the osteoblasts, human derived. We are also trying, no animation, no pictures, sorry. Here. We are trying to use not only 2D model, but also 3D model in the matrices, which was very, and that's why it was very inspiring to hear the lectures from Maria and Maria to see the possibilities also of bioprinting. So far, we are using the model in the matrix, which is not bottom up, as mentioned by Maria, but top down model so far, and we are having the material in the 3D matrix. My PhD student is now working on it in Berlin at this short-term scientific mission. We are also trying to use the perfusion system in this model, and we are trying to bring the composition of the media similar to the composition of interstitial fluid, which is in contact with the implant, but it's not that easy because the composition can vary a lot of the interstitial fluid, and we have found out so far that there can be many differences in the different media in terms of the corrosion of the material as well as the cytotoxicity of the whole setup. So now we are working mainly on the media and comparing the media. And there is also the question of the ethical question, and it was already mentioned by Emmanouela, and it's because it's very ethically problematic to use fetal bovine serum, and we are also trying to or planning to somehow replace it. And interestingly, we were we were planning to go for Panexin as well, but we will consider seeing these problems with it. Yeah, that's it. Sorry.
Okay, so thank you. I would like to ask all the speakers of this sub-panel to join us now on stage, please. So it would be Joana, Maria, and Eva. So for questions, please do submit them through our QR code. And let's start with Joana. So your presentation had like quite a lot of questions arising. So let's start with, did you taste the print? Do people find them tasty? And what's the consistency like?
Well, the smell was a problem. So, but we didn't like reach that part of the investigation. It was I was doing this for the first time. It was in my master thesis, so we started very in the beginning. So I didn't have the time to get there to try and with showing people to people what I did and to have a feedback. But for some colleagues, I received some feedback about the smell.
Well, I was going to say, that's a shame, because the pictures certainly made them look like a fancy box of chocolate. So kind of, I guess, along that theme of at least what they look like, there's been a question about, should we be printing food based on these diatom shapes, or would it be better to print based on, I guess, what people are already familiar with?
Well, I think that diatoms, if we can put in context and talk about the sustainability of that, and because, like I said, they can capture carbon, and they're environmentally important. So I think if we put it in context, it should it should convince people.
So I guess it's more about the story behind it.
Yes, exactly.
Mm-hmm. And I guess another closely related question, and this comes back to testing them. Do you know if there's any concerns around the food safety associated with 3D printing and food?
Now, if we, in the conditions that we did it, probably there's some concerns, but if we have if we follow all the security measures, and we can have 3D printing structure that can be secure. So, yes.
Yeah, that's a really interesting topic that we could further discuss, mainly due to the nozzles used in 3D printing. But now I would like to ask Maria, do you think AI can complement at any stage the biofabrication process, namely in your case study?
Yeah, thank you. Yes, absolutely. Thank you for this interesting question. AI has been already integrated in many of our stages of our work regarding the prediction of the properties of a biomaterial, but also the design of a model, and of course on the effect and the activity of the drug as well, or even the selection of the drug. So, yes. Absolutely. Machine learning and artificial intelligence are already incorporated in these kinds of multidisciplinary topics.
Yeah, and it's nice to see that we should learn how to use those type of tools to further help with our studies. But moreover, people wanted to know, how does biomedical research on cancer contribute to nature-based center biodesign?
How does it contribute to nature?
Nature-centered biodesign.
As we heard already from this morning, the selection of the processes, the selection of the materials can be naturally derived. This is one aspect. We can see that nature-centered or naturally derived materials can serve as best candidates regarding biocompatibility, but also processability, manufacturability. For example, the polysaccharide, alginate, and many more that are derived from organisms. They can be low cost, but also their properties can be very useful, and we exploit them. For example, the high viscosity is one of the rheological properties that we exploit in extrusion bioprinting, for instance. So there are many benefits and many properties that we can exploit when we focus on naturally derived materials. And excuse me if I may also add, nature center is another direction. We and other labs also work on dynamic cultures, which mimic the in vivo situation. I haven't shown any data today due to the lack of the time, but this is another aspect that helps us minimizing the in vivo animal testing, which is very, very important as well.
Thank you. Eva, do you have any additions to that around kind of the biomedical research and bio-centered design?
I also see the main issue in 3R principles that we should somehow prefer the advanced models which will not use animals or animal-derived products, and we should do more new approach methodologies which also are 3D printed or 3D structures and so on. Yeah.
Yeah, so there's a lot of cross-learning that can be had from, I guess, like how you're 3D printing things to how we 3D print food, as there'll be some similar challenges there. So another follow-on question based on your talk is, can materials be living instead of biodegradable in order to perform the beneficial actions to the bone?
Sorry, could you repeat?
So in your talk, like you're obviously talking about beneficial actions to the bones. Could you use living things instead of biodegradable materials?
Living things like
yeah. Can these materials be living? So I guess active, like living cells, for example.
There would probably be a problem with tissue compatibility, but yeah, I probably also. But we are mainly focused on biodegradable metals, which should be they should have like proper mechanical properties and so on. So yeah.
So I guess if we found a living material which had the correct properties, it would be something that could be experimented with if you got past the ethical side of that.
In case of bioprinting, we use already living cells inside the ink. Mm-hmm. So a bioink plus living cells is already a bioink or a bioprinted construct. And in these constructs, sometimes we add fillers, nanofillers, like metal ions or bioactive glasses, whatever. So biocompatible components which can be used in order, for example, to improve the mechanical properties as well. So many of these materials can be combined with living cells as well.
Mm-hmm. Thank you. And so another question for Joanna. Could biases towards negatively valenced animals in conservation extend to reducing food aversion and supporting interdisciplinary translation into practice?
Sorry.
So could
I think the most biological way to put this question would be something like, could the bias towards like the animals that are going extinct extend towards reduced food aversion? So, for instance, in terms of the eating the gametes.
extinct. What was?
So the question literally, it's like, could the biases towards negatively balanced animals in conservation extend to reducing food aversion and supporting interdisciplinary translation into practice? So I think, and if whoever did the question is present, could further extend on that, but I would say from my interpretation is that if you could maybe utilize that as an explanation to why you should use gamma rays.
Yes, sure. Because gamma rays can be created or produced in control ambient environment. So they produce it at the EcoMar. So we can use that and prevent some of that issue that is the possible extension of some species.
So, and you touched on a rather interesting point, because we do produce them at EcoMar, and we do use control conditions, but there's also a question if you have to control the conditions in which you 3D print the food so that you can keep the nutritional value of the, I guess, cookie.
Yes. The printing paste the paste is printed at room temperature. So and then when we freeze dried it, it's safe. So in terms of 3D printing, the 3D printing itself, I think there's no problem in control conditions, because the control conditions that we had is just room temperature and
So there is over here a last question that I'll ask you, and then I will release you all to lunch, I think. But do you believe that the ugliness of the amphipods reduced the exception rate? And to that, I would like to add, do you think that if we call them like seafood cookies or something like that would be easier for the common perception?
Maybe, yes. Because the appearance is the main problem, but we grind it, so when we put it in that structures, we can get that cookies that if we say that it's seafood, because it is seafood, and although it seems like insect, but it's seafood. I think it could generate more acceptance.
Yep, that's great. So can we thank our three speakers from this second panel?
Thank you very much. Thank you to the speakers. Thank you to moderators. Thanks, Victoria and Ruben. So we will have lunch now. We were a bit delayed because there was some stuff with the restaurants, but all is fine. I'm just going to ask you to be here again at 2 o'clock as opposed to 2:30. So you still have plenty of time to, have a nice, calm lunch and enjoy the sun. Again, so for the vegetarians, please follow Pierre. And for the restaurant, buffet, where is Gonçalo? Gonçalo is outside. All right. He's already there. All right. All right. See you at 2.
Through the hall?
That's
why I was in Miracle.
It depends on people, really. start? Shall we start? So not everyone's here, but I think we can start already with the third panel dedicated to computational design for computation for biodesign. Yes. So the panel is run by two moderators, Branimir Pavlić from Serbia, University of Novi Sad, and Renata Troyan from France, Institut Supérieur de Mécanique de Paris. Okay.
Thank you, Nina. Okay, so I hope you will not be sleeping after the lunch during our panel. So we will try to make it more interesting possible. So we will start from now. I will begin the panel by presenting myself, but in reality, it's my colleague who submitted the presentation, Monsef Hamadi, who developed the method and who invited me to join him in this adventure. So as we are working in the engineering school, we are teaching the future engineers, and the objective of this approach is to teach our future mechanical, mechatronical engineers the nature-centered computational design making, in hope that they will apply it later when they will work in the industry. So the approach overview in a couple of words. We are starting from the design problem. We evaluate the design problem, what are the requirements, what are the constraints, what are the objectives. We apply several steps of design principles, trees design, inventive design methods, nature-centered design, calculating Pareto front, with the objective of to select the robust solutions that stay inside a safe operating envelope. And how can you imagine? It's really nature-centered solutions. What the words that we are listening all the day today, it's robust, balance, adaptation. So the solutions that we want the students to obtain, they have to be balanced, they have to be viable, and they have to be robust. How do we do this? I present each year we teach the special model, it's in French, so CBO, we'll say, and we propose them one test case. So here you saw in the morning the robotic arms, who's very interesting, and the classical of mechatronics design. We start from the analyzing the problem. We analyze it. What are the contradictions that preventing us to obtain the perfect form? What we want from this design robotic arms. We want it to be lightweight, because when it's light, it's easier to manipulate. We want it to be precise. When it's very light, it's easy, to vibration. It's really hard to center. When it's heavy, it's more easier to make it precise, but it's harder to manipulate. It demands more energy. So you see here the contradiction. How can we solve it? First, we formulate the ideal final result. It's coming from trees, inventive method. What it would like if we had the magic wind, like in Harry Potter, and we can apply it, and we can want the perfect robotic arm. It would be totally lightweight and perfectly adjustable, maybe like our arms, something like this. So first, once we formulated this, you can see after this ideal final result point on this screen, we study the nature examples. What are the ways the nature is doing this? So here you can see, for example, the elephant trunk, elephant trunk, or the tree trunk, or the head of the bird, or maybe the muscles. So all these structures have the useful points we can apply for the design of these robotic arms. Once the students have studied it, we choose the two contradictive objective functions that we will find the compromise. How can we modulate them? So for example, here in this example, you have the structural weight, so we want to minimize the weight, and we have the precision, so reducing of the vibrations. What we can do? We modelize these robotic arms, we make the calculations, and so we can, with some optimization methods, classical one, we plot the Pareto front. So Pareto front is all solutions that each one is not better than another one in both objective functions, both criteria in which we want to optimize it. And here a small star is the ideal results that we want to get to it, the utopia point, you know, that our regenerative solution. So once we have this Pareto front, we pass it through several filters. So first we study what are the not realistic designs. So we can make it on the simulations, but in reality we can't fabricate it. We can't manufacture it. It's not passing the constraints. So here you see blue one points. They are not good points. They are really cute, but they are not realistic ones. The red one are staying. Once we make these hard passes, hard filters, we are going to the balance score. So here it's more to see the viable solutions, like nature imposed solutions, the solutions that are sustainable when we have some changes of operational areas. So the nature, it changes more the robust solutions that can operate if your operational conditions are changing, then then something that is perfect in some specified conditions. So once we have this graduated Pareto front and the solutions that are coming from the nature, from the relevant examples I showed before, usually they are here in the middle, the yellow color, that are perfect, the best solutions. Once we do this, we pass in another one filter, the real applications for mechatronic design. And here you see we are going from the blue one points to red line. The solutions may be worse, but they are more robust, meaning that this solution is more reliable in the lifetime of the structure. So we passed all the filter, and we obtain the perfect solutions that we can choose. But as it is computational methods for group, so all this is automized. We use the computational software. So first we program our design system with a system modeling language. After we making different simulations in Modelica software, we choose various scenarios. If the simulations are too hard, we can obtain the surrogate modeling. It's approximative modeling that allows us to have the short simulations for the similar problem. And we can construct our Pareto front. And like this, we obtain this flow work that is automized. So for the first application, if you need some updates for your robotic arms, we can make it there with a project with our students and to see what can we do make more. And as a conclusions, with our ideal final results and Pareto variability balance provides filters, it provides a teachable computational driven path for students, and it's possible to apply it for modern engineering companies. And the most important message to send to our students that you have choose the most resilient options, not the best nominal options that looks cute. Okay, thank you a lot for your attention.
Thank you very much, Renata, for opening this very diverse section with an interesting topic. Now we proceed further with Gabriela Madeira Ramos. It was okay.
How do I? Okay. So hi, I am Gabriela. I am a PhD student here at Aveiro. I work in ID+, and TEMA, which is a collaboration with the engineering department, because I'm a mechanical engineering PhD student, but I came from product design. So my PhD context is renewable energy projects, infrastructures, and basically those projects are very huge, and they answer to an urgent need for better energy with better resources. from the environment, because they apply very diverse impacts, namely in environmental context, but also with communities, so social impacts also, and even within the technical part of the infrastructures too. They are big challenges, those types of projects. There is, although there are a lot of sustainable guidelines for those types of projects, there is a lack of design guiding those projects. So how does those clusters that already exist come together and work together more efficiently towards a sustainable goal? However, during my PhD, I have found myself arguing about that sustainable context or concept, because if we are using renewable resources from nature, why not go beyond sustain them, but regenerate them? Because if we need them further, we need to also care for them. So that's how I'm trying to develop a new way to evaluate and a framework to evaluate and also guide the design process behind it. So this requires a shift in the measurements also when we do this shift for regeneration, because it's not only measurable criteria that we encounter, it's also very qualitative, because nature sometimes, or even us, we don't apply only qualitative kinds of measures. So that's why my framework, I have gathered data on several different scientific areas that seek to define what regeneration means to them. And from those frameworks that already exist, I have come with this one that combines into 4 domains, which is the design impact, the place, firstly, the quality of life, design impact, and the progress. And they go from the nature scale of it, so what already exists and what do we need to preserve, and to societal needs, going from ecosystem scale, so what is the quality that we need to achieve for the ecosystem to live, and also what is the human scale for communities to grow. And then get a measure for the progress that it's going to happen, because the main goal is the co-evolution. So the learning process is also very needed. And therefore, I have also tried, because we're talking about gathering engineers, but also designers, but also communities that have not a very big notion of the projects. We are also gathering biologists and ecologists. So this means that the design process of it is not always tangible and is not always abstract either. So we need to tackle those differences. So I have come with 48 indicators that are mainly qualitative for now, and they go from like operation location, heritage. Like first, we really try to see what already exists before any project should come about. And then we try to basically evaluate what is the current quality of life or the living requirements that there is, but also the historical one, before even we had the idea. So what has been lost and what can be restored. And then there's like the design impact that goes around it, which also takes like this planetary world view, which is already like a big thing, because in those projects, the designers have a little voice mainly. And so, because it's very technical centered, like changing the mindset is already something quite big. So we also tried to create like this positive impact with the requirements that we have gathered before, and also think on the long term. So operation and maintenance should always be present, and also try to balance the mediums that we use with the design process, because there's stakeholders that are very different. And then there's the progress domain, which basically not only quantifies the growth, quantitative and qualitative always grouped, but also like seeing in a planetary scale what are the boundaries that we are trying to reach, like the low boundaries and the higher boundaries. Where are the projects in those contexts? And also, like I said before, the coevolution and the learning is very, very important, because projects such as the energy systems, they live for at least 40 years. So it's a long way to go. Sorry. And then, because like I said, I have gathered a lot of qualitative qualitative indicators and measures, but also I need to tackle the more quantifiable indicators. So I thought, why not try to apply what already exists and is quite implemented, which is the life cycle assessment. So I have also spotted some potential collaborations and you know, get the qualitative data and the quantitative data to work together as a final score. So this is what I have for now, which is I would say it's a beta version. Which already gives us, this was like a case study that I did, and it already gave the final score of the project. So that would be how it would end. So the project team would have access to this platform and would enter those datas, and the datas would be codependent within the network that is right here. And they would also be guided towards which design strategies could they apply, because the stakeholders are spotted, because there are the scores. So each indicator has its neutral score, sustainable score, and then regenerative score. And so they would know which strategies to go beyond the level where they are at now. So ongoing and further work, like I said, is to integrate further the LCA indicators and also maybe social indicators, because there is some indicators that already exists, and seek to incorporate also better qualitative and quantitative data together. This is where our collaboration is very useful for me, and for the work. Because I'm a designer, I'm not always I'm not a biologist at all, even though I really like nature. And also try to see, instead of going from regenerative potentials, which is what I have now, I see the potential paths to go. Why not see regenerative trajectories? So maybe for each independent project, have them actually have like a step-by-step trajectory and not just a whole calculation of the final score. And yeah, basically this, and also testing empirically without just theoretical case studies, testing with designers and engineers and biologists. How do this tool could be as much useful for you as for designers? Yes, thank you.
Thank you, Gabriela, for sharing with us this unique approach aiming to bridge differences among various interested groups. Now we proceed further with some predictive modeling for the natural forms. I invite Elena Hajieva. Please, Elena.
Hello from me. Thank you, Branimir. I'm a mathematician, and I will talk about mathematical constructs that approximate the natural forms the best. Those are such called fractals. They are not in the title, but when we say generated by iterated function systems, we mean on forms or on fractal forms generated on this specific way, because there are many other ways how fractals can be generated. Maybe you have seen some fractals, but they are only colorful schemes. This type of fractals can really resemble some natural forms and processes. Some of the forms that exhibit irregularity, complexity, self-similarity are shown there, like coastlines or plants like broccoli or fern. And these objects are such that cannot be described with classical Euclidean geometry, lines and circles, triangles, rectangles, but they need some more complex structures for descriptions, like fractals. This way of fractals is just representing them as a set of points by means of a finite family of mappings which iterate the beginning, some beginning set or point, whatever, and they converge towards a form that exactly exhibits complexity, regularity, or fragmentation, just like natural forms are. That's why I choose this topic today, because these forms have potential application into biomimetic design or regenerative design, or maybe ecological system thinking. But of course, we may discuss after this is just general presentation, we may discuss and try to join our forces. As the title says, we have a way to predictively and intuitively model or transform the fractal forms in real time, and that tool, its main window is represented here. It is called Fractal Engine. For now, it works for two-dimensional generated fractals. This is an example of fractal that is called Maple Tree. Some functionalities for coloring fractals are shown. The tool works such that first 3 points are pinpointed, which are called control points, and by clicking and dragging the points, I can show you the application later, whoever is interested. By clicking and dragging the points, the form of the fractal is changed, which includes different types of transformations, rotations, scalings, reflections, translations, but also there is a possibility for more complex transformation, like cubic transformation, quadratic, or exponential. There is a possibility for better control over fractal modeling when those 3 points contain, I mean the triangle may contain the form, then the control by means of these 3 points is better and ensures the convergence of the process. The tool has possibility for animations, like you have initial form and final form, and some blending between 2 fractal forms can be generated and also exported as a GIF file. This also, and frames can also be kept, which enables explorations. So what are the forms in between? Or maybe what I can assume as an application if you have an initial form of a system and final form of some natural system or phenomena, maybe the development can be catched with such approach. There the cubic transformation is explained, maybe not so useful, but again, in the exploratory sense, one can explore different fractal forms. And just shows some of the functionalities. There are many functionalities, but also many space for adding new functionalities. This was about modeling fractal forms. But the bigger problem is how to create them. So if you have some objects from the nature, how to find that iterated function system, that family of mappings, to create the target form. Theoretically, that problem is solved because there is such called collage theorem, which has rigorous mathematical proof. Everything works perfectly, but when it comes to apply this to create some natural object, there are some limitations of this theorem. The first one is that heuristic procedures are required. Actually, some experience is required from the creator, which is obtained either with working in mathematics for a longer time or with trial and errors, how to create those families of transformations. The second one, I can't explain because I can't explain now because I didn't go into details, but in general, it's about the collage. The collage is on the upper left image. So if you want to create a tree, you can do some contractions of the overall rectangle to describe like a very basic description of the object, and then to repeatedly iterate the process, and you will get the form on the right, which looks like a tree. But the form to the right, that tree, shows perfect self-similarity, meaning that each branch looks like the whole tree. Then I come to the third limitation. In order to get more natural objects, like with not all branches to be the same and to resemble the general object, we have to add some randomness of the maybe angles of these branches, or maybe contraction factors, so some to be smaller, some larger. For that purpose, the system might need infinitely many mappings, constructed on a random way. For help in that direction, we may need AI tool that will make maybe trial and errors with human help until we reach a form a form that we need, a target form, and that can be a future work. I wonder if there are computer science engineers here or programmers, because in the list of disciplines that Gabriela mentioned, she noted that perfectly. But maybe there are no such from such fields. But if there are, maybe we can talk. And also, there are the future work can be extended to improved functionalities of this existing tool, extension to 3D fractals. We already have one for 3D fractals, and they might be integrated. That is also possible, but also this tool, which I just described, can be upgraded itself. And maybe this is the most important, AI-assisted discovery or construction of iterated function systems that generates natural objects or systems or phenomena. That would be all from me.
Thank you very much, Elena, for sharing with us a topic with a huge applicative potential. Now we move further to the topic from plant growth to additive manufacturing, and I invite Elisa Mazzoni. Please, Elisa, the floor is yours.
Thank you for the presentation. Yeah, my name is Elisa Mazzoni. I'm an architect and a PhD student at the University of Florence. The research I presents the results of Muralis study that is part of my PhD research conducted in between the University of Florence and TU Delft, and which investigates how additive manufacturing integrated with parametric design can enable multi-species technological network-based solutions at the facade component scale. Despite the growing interest in AM-based vertical greening systems, vegetation is often treated as an applied layer, prioritizing human-centered benefits. The Muralis study addresses this limitation through an interdisciplinary research by design approach, prototyping a 3D printed clay facade component optimized for the integration of this special plant that is Cymballaria muralis. That you can see here in this photograph. It's a stress-resistant plant typical of the Mediterranean climate. It grows spontaneously in the urban ecosystem and is naturally adapted to growth on artificial vertical surfaces and resists to urban stressors like high surface temperatures or prolonged water stress. That's why we choose this plant for the as a case study for the design of the component. So acting as an architect, as I am, the first step was to understand the needs and behavior of this plant and try to translate it into parametric schemes able to inform the geometry of the facade component. So in this case, you can see upright, the growth action scheme, and you can see the direction of growth transformed into vectors, and the anchorage points into points, or bottom left, the leaves distributions transform into surfaces, and then the solar exposition and the shading needs of this plant. Those parameters were then translated into the geometry, and here you can see the final prototype, characterized by an horizontal development, a shaded space for the central root system, hollow volume for inserting the substrate, and another characteristic is the punctuated, perforated surface created with in the grasshopper environment during the parametric design process. We create this pattern with wavy layers that let us create this perforated surface for rainwater penetration and the sprout growth. The openings for water drainage, that is very important. Here I will show you a short video that explain the process that follow the additive manufacturing phases, starting from the 3D model creation in the grasshopper environment, passing through the 3D printing with clay. In this case, this is the liquid deposition modeling process. Then we will pass to post-processing operation, like control the measures, firing, weight, and then this is the validation phase of the geometry. And we decided that the only user that could validate the geometry was the main user of the tile, so the Cymballaria muralis itself. This is the first test with cuttings of Cymballaria, a mature plant of Cymballaria muralis. But then we discovered that the perforated surface, the perforation were too small to insert well the roots of a mature plant. So we decided to did another test by planting directly, by seeding directly the substrate. And here you can see a comparison. I took photograph each 15 days, more or less, but here you can see the 19th of September, the first sprout. And then in November, you can see young plant of Cymballaria muralis there. And you can also appreciate the difference, for instance, in chromatic differences and the bioreceptiveness of the tile. You can see some green up there. Those are microalgae and mosses. In conclusion, I want to show you the second result of this experiment, that is this design causality map that put in relation all the parameters I took into account, so architectural parameters, fabrication and biological parameters. And that's quite, even if it's very specific of this human-driven experiment, it's interesting because provides a qualitative representation of influence and design dependencies, illustrating how changes in one parameters can trigger all the others. And yeah, for instance, I don't know, increasing the hollow volume can let us to put more substrate, so the plant have more space to grow in it for the root and so on. But increase also at the same time the weight, the transportation, and the structural reliability. Thank you.
Thank you a lot for your presentation. So the next participant, Václav Sebera, who you will see maybe some analogy with the previous presentation about the inspiration from the plants and roots.
Thank you very much. Hello, my name is Václav Sebera. I'm coming from Brno, from Czech Republic, and I study actually wood. I'm wood scientist, but we also have a few projects on trees. And this particular presentation is on the underground of the trees, so the tree roots. Yeah, actually, as we know, arts and sciences have a lot of in common, and not only in principle, but also in models and actually the objects they study. So you can find trees in many paintings, contemporary paintings, of course, old ones, or you can find the sculptures like Giuseppe Pennone that has very nice installations of trees, or you can find it in many scientific publications. I chose very known one from Charles Darwin. This is actually the only figure in his book, and it's a phytogenetic diagram. We call it tree diagram. So our intention at the department, at the project is, can we get inspired by trees furthermore? And that leads to my title of the presentation, what about the tree roots? Can be inspired? We already know they can, and I will show you what we do. We do test the whole trees with soil or without soil, so these trees of course die afterwards, but they are meant to die nonetheless. So this is what we, for example, tested in Croatia, in Zagreb, and the pine from Kaunas, that's from my walk, that's not from testing, but you can see that the tree has many different strategies how to survive in nature. Of course, the conditions on a site decide if there will be a lot of fiber and fine tree roots, or there will be like very little tree roots, but of main support. So we have a tap root, and then we have a supporting roots to the side. So of course, tree has to meet these requirements: the physiological, the water transport, and the mechanical. That's actually the evolution of trees. So trees are very effective in encouraging, because they have a huge surface area to volume ratio. So if you take 1 cubic meter and calculate the surface of the trees in 1 cubic meter of the tree root, it's way, way higher than the conventional man-made foundations we do, like using the micropile foundations that are actually the most advanced. They have the biggest surface to volume ratio, or common foundations of family house. And luckily, actually, we are not the first who wants to get inspired from tree roots to think about a new foundation system. There are already few publications that think about it. So I think the Stachev et al., or there's a group in France I know about. So they are thinking about these telescopic tree roots that could actually go through the soil and provide these necessary transfer area to the soil. Yeah. So our little project consisted, our actually first step is to understand the interaction of soil and tree roots. So we took one tree that is actually on the left, on the top. It's a beech tree, Fagus sylvatica, and we did pulling tests. So we pulled the test from four world directions, statically and dynamically. Dynamically means that we will pull it and we just release it and we analyze the vibration of the tree. Then we did it without the crown. So we cut out of the branches, and the third stage was remove the soil at least to 30 centimeters. So that's what you see on the picture. So we have many measurements from one single tree. Then we excavated the tree. That was very meditating a part of the job because we were laying down there for a whole day in 3 people, and we tried not to break any, let's say, significant root. Of course, then we cleaned that. We had to remove the fine trees because of the scanning, and we wanted to digitize tree roots, so these fine roots would actually result into very computational expensive models. So then we laser scanned and digitized. Of course, we had to clean. The roots actually are very porous, and they are also very, let's say, heterogeneous. So the model you see is already smoothened to the very big level. So you see how it, how it's smoothed. And then we transferred it to the numerical simulations using finite elements, or now we are starting with the discrete elements also. So we have models. On the right, you see actually models from both, from the discrete element method and from finite element method. And now we examine the interaction, because there is a big problem how to actually model the interaction between soil and root. What is the friction? How actually can we use the friction to simulate these fine roots that are actually removed from the model, etc. But we somehow can see the similar patterns in behavior in the different 4 directions, like we saw in nature for the tree, and now we are actually in the middle of processing of the data. So there are not many projects yet, no results yet, but we hope that we will get it this year. So that's all about the tree root project, and on the end I show you what we do also at the department, because it's maybe connected to the fractals things. We also do the trees that are on the right. So we either use the generation using L systems, which are based on fractals, or we have beam models of the trees using the scanned data. We do, of course, the composites or the structure of wood, either generated probabilistically or modeled using CAD. And if we are bored, we cooperate with the people from musical industry. So we have a good cooperation with a Czech producer of pianos, Petrov, and one guitar producer in Czech Republic. So that's all from me. Thank you very much.
Thank you a lot for the presentation. So we had mechatronics, mechanical, designer, architect, mathematical, and now chemical engineering. So the last presentation today, it's Branimir will present.
Thank you very much, Renata. Hello, everyone. My name is Branimir Pavlić from the University of Novi Sad, and I'll share with you some of our solutions for the optimization of isolation of bioactive compounds from natural resources. So first, let's see the roadmap of the scheme and the project that we recently had. This is the 4-step innovation pathway from the raw materials to novel products. And in each of these steps, there's huge possibilities for the innovation. Namely, first, there's a lot of potential in the novel and sustainable resources. So we could find and utilize underutilized crops or food industry byproducts, agricultural waste as the potential source of bioactive compounds. And for that purpose, we could try to utilize green extraction techniques and methodologies in order to improve the process and increase the yield of the target compounds. And not only the yield of the compounds, but also we can modulate the bioactive potential of the obtained extracts. And in third step, there's a potential for the optimization related to the maximizing the yield of the target compounds or targeting the specific bioactive potential and also the possibilities of determining the relation between structure of the compounds and the effects, biological effects that they are producing. And finally, the huge potential is in the development of the novel products. Novel products specifically for the food, pharmaceutical, or cosmetic industries. So why natural deep eutectic solvents? So even though we are familiar with the eutectic phenomena for more than 100 years, there wasn't no large application of this phenomena throughout the last century. The real breakthrough was the study by the group of scientists from the university from Netherlands, where they hypothesized that in the living cells there's a third type of liquid. Not the water phase, not the lipid phase, but the third one, which can explain the solubility of bioactive compounds, of biomolecules, that can explain transport phenomena that occur, that can explain biochemical reactions that occur in the living system. And from that moment on, that opened the gate for the application of this type of solvents for the isolation of bioactive compounds. And immediately they confirmed that there's a tremendous increase in the solubility of some bioactive compounds comparing to the conventional solvents. For example, in case of rutin, which is very famous flavonoid natural compound, there's a tremendous increase in solubility comparing to water and when you try to dissolve it in the eutectic systems. And not only the solubility is the advantage. With biomolecules, we have a lot of problems with the sometimes or quite often very poor bioavailability when we ingest them in our system. So even though we had high concentrations, the bioavailability is still too low. So a lot of studies already confirmed that we can improve bioavailability tremendously with eutectic solvents. And that's the reason how they affect transport phenomena to our semi-permeable membranes. So what are these eutectic solvents? They are made with natural compounds. That's the first and the most important aspect of them. And we use hydrogen bond acceptors and hydrogen bond donors. And you can see that hydrogen bonding is the major molecular phenomena that occurs within this solubility. Not only the hydrogen bonding, but also lipophilic interaction, some van der Waals forces. So weak type of molecular forces. However, in that way, we get solvents, natural solvents with very unique properties and high selectivity towards a lot of different bioactive compounds, no matter the hydrophility or lipophility, no matter the size, even from small molecules to very high macromolecules. And that means that they have a lot of potential to be used as solvents for the isolation. So I'll show you one case study, one successful case study for the valorization of wild-time herbal dust. So the herbal dust is the powder plant material obtained as byproduct from processing of filter tea. It's a tiny powder fraction which can be used as filter tea in tea bags, and usually it's produced in 10 to 20% during the production, so it's not negligible. There's a lot of huge quantity of that byproduct, which is a very good raw material for the isolation of bioactive compounds. And this is the suggested three-step optimization approach. I'll go to this quickly. So in the first step, there's all parameters are constant. We use only screening on the type of the solvent, eutectic solvent mixture that can be used. After we determine the one or two with the most promising potential, we use the factorial design study to screen for several factors, in this case, 5 factors, which could affect extraction yield about the most influential factors and their experimental domain, and we can set the optimization study. And after we do the experimental work, there's some predictive modeling. In the second phase, there's linear and interaction models. In the third phase, there's quadratic models. And if we get good fitting of the results, we can predict the conditions for the maximized yield of the target compounds. In this case, 5 response at the same time using a desirability function. They are maximized at the same time. And of course, you can get calculation of the optimized extraction conditions and experimentally check in the end how does it fit with the predicted results. In this case, it was very efficient. And in each step of this optimization study, there's a huge increase of the target compounds yield. For example, from 1 to second phase, there's a 20 to 30% increase in polyphenols yield. From second to third phase, there's also 25% of the increase. So from the initial step to the last, you get a huge improvement with your target compounds using this approach. And when you compare this data with the conventional extraction methods, the results are even more terrifying because you get at least 10 times higher yield comparing to the conventional solvents such as water or ethanolic mixtures. So this is one example of the successful utilization with the high content of rosmarinic acid. Rosmarinic acid is a very potent bioactive compound with lower bioavailability when it is ingested as a food or as a supplement. And now we are working and we have some preliminary experiments how we can utilize this type of extract with eutectic solvents in order to improve its efficiency in the functional food products. Thank you very much for your attention. We invite all presenters from this section. Was I on time?
No, we are not in time at all. Everyone spoke 10 minutes instead of 5, but we have 10 minutes for questions. The questions are very, very interesting, so I promise you we'll follow these questions for the people who are presenting. Oh, it's okay. Okay. We are 2, yeah, we have 5. No, it's okay, it's okay, Nina. For 10 minutes we will stay, yeah. So, yeah, we have one. No, it's okay, Nina. Okay, maybe. So, to go fast. So, the questions for Elisa. Can the plant validate the prototype? Because the plant has its agency, and so, what do you think?
So if, sorry, if can, yeah. Yeah, I mean, it's what I've done more or less. I mean, I started tries or the validation from the biological component, in this case, this plant. So, yes, that's what I would like to do. I know the experimentation is at the beginning, so we had the chance to monitoring it only for 2 months. So, but yeah, I hope to work on that in the future.
And the question that is a little bit similar about the agency of the trees, maybe, so for Václav, are there ethic approval to perform this intervention in these trees?
Yeah, trees have their own rights. We know that. It's in Czech jurisdiction. I think every country has some law on tree let's say, safety and also protection. But we work with trees that are going to be cut down. So it's already decided by the institutions of the owners. Sometimes it's the municipalities, they gave us trees because they want to build a parking place or something, you know. These crazy things happen, yeah.
Okay, thanks. So they are not just dead, they are useful for the science.
We don't have to do any ethical, let's say, paperwork for the trees. It's not like with other materials. Yeah.
Okay, thank you. Yelena, does the function follow form or form follow function?
Well, if one wants to make the target image by iterated function system, that means function follow the form. But maybe the other way, form to follow the function happens when you want to explore some new IFS code. So you're changing some number in the the functions are formed as with matrices and vectors. If you change just one coefficient, you can get some unpredictable form. So the form and also the location of these fractal images are not predictable. So we can go two ways. Direct, such called direct and inverse problem.
Thank you.
Okay, there's several questions for Gabriela, but since we are lacking time, I'm gonna randomly select one. So, Gabriela, did you notice a need in any industry of a real case that prompted you to develop qualitative indicators?
I need you to repeat it. I'm sorry.
So, did you notice a need in any industry of a real case that prompted you to develop qualitative indicators?
If I found in
what was the motivation? Some industrial partners asked you to do this?
Oh no, yeah, I mean yes, yes, it did not pop out of my imagination like that. But initially, I started my PhD with a company for renewable energy, but it was not in this regenerative direction at all. Afterwards, of finding the process of trying to find how information on how actually energy projects are done, at least here in Portugal, was kind of tricky, and it got me really to think differently on other ways to see, because it's a very technical field. So a lot of, and I am a designer, so a lot of information that I got was on the energy efficiency, and it's a fact. It's the biggest impact with energy losses and everything like that. But then in the last couple years, the European Commission has gathered some guidelines on including community into those projects, and including also like having biodiversity targets also, and not only on energy system projects. And I thought, why not gather, the two fields together? And so by that time that I figured that out, the energy company here in Portugal, like the project was already ended, and I did not have kept contact, because they were not interested in that. However, there are other companies interested, namely, I don't know if I can say it. Stedin, okay, I didn't know if I knew about. Namely in the Netherlands, which I'm going to collaborate in a very few weeks. So yeah, basically.
Thank you. And one question for Renata. With the growing application of AI, does it replace entirely the process you showed by automatic generation of data and ideas?
So, thank you for the question. The answer is in two parts. First of all, AI, it takes a lot of energy, resources, so if we can do it normally, let's do it normally. We know how to do it explicitly, we understand the way, we know the rules, so it's working very well. The second part of the question, when you ask the AI to do something you don't know how to do, it gives you really good results. When you ask it to do something you are a specialist in, you see a lot of mistakes. So no, it can do. You can see images during these presentations all the day. In each image there is something that is going wrong because AI generated it. So for now, no, and we have no need to do it. It's okay for now, like this.
Thank you.
So do you want to read your question? Because your question is so complicated, so I'm not...
It's a bit. It's a bit complicated. The author maybe can clarify, but it sounds like this. So, may you expand the impact of these results for the biofabrication of enhanced phenotypes, such as targeting warfare or resource engulfment? Okay.
Yeah. So, in this world where we are facing towards a sci-fi type of technologies, and in this political era where a single button can trigger a warfare in a place, what if we can change the components of a cell, replacing the entire function of it by a faster way of making DNA replications, proteins, that can lead to enhanced bodies that can be instrumentalized elsewhere?
Yeah. I'm not sure if the eutectics can reach such as ambitious breakthrough. Basically, there is no restrictions for the application itself. I mentioned it's made of fully natural compounds, and what's good here for majority, if not all of these compounds, they are quite safe to use, and at least we have permitted daily intake information or safety information about them. So application can be broadened tremendously in case also of preservation of some biomolecules, including some very sensitive biomolecules and the genetic materials. But I'm not sure if they can speed up some genetic manipulations itself. It can only improve the process, but I highly doubt that it can be speed up effectively.
Yes.
Thank you.
Of course.
Thank you a lot. Thank you for the presentations, and do not hesitate to discuss later.
Thank you. Thank you, Renata. Thank you, Branimir. And thank you to all the panelists. Now I will call... just one second. All right, yes, okay. So we are in panel 4. There will be 2 sub-panels here. The first one is biofabrication, advanced materials and infrastructures. The 2 moderators are Jelena Brašković, an architect from Serbia, University of Belgrade, Faculty of Architecture, and Adrien Rigobello, a biodesigner from Denmark, Royal Danish Academy. Welcome. Please.
Just to greet you from our side as well, VG4 should gather knowledge from all previous panels and all previous working group, and will focus on application and interdisciplinary collaboration and application, yes, of bioregenerative research, and therefore we shall lose time no more, and we We'll go to the first sub-panel that will be moderated by Adrian. It's about biofabrication and...
Yes, thank you, Jelena. So, hi everyone. So, indeed, biofabrication, advanced materials and infrastructure. So, we have 3 presentations in this first panel, so that should cover about these 3 aspects. The first presenter that we welcome is Jelena Jokic, excuse me for the pronunciation, assistant professor at the Faculty of Mechanical Engineering in the University of St. Cyril and Methodius in North Macedonia. Welcome.
Thank you. Thank you all. Hello, everyone from my side. So I will present you a little bit something that we are working with my colleague, Assistant Professor Elena Angelesca, and my master student, Tamara Stefanovska. So basically, before I start, I would like to show a bit background of what I am working. So I'm industrial designer, but working in the field of mechanical engineering. So mainly, I'm interested in finding solutions, finding application for some real life problems. So what we are apart from my research area is working in finding inspiration from nature and implementing it into product design, parametric design, and computational design, because also I have background in additive manufacturing, so I'm exploring how the possibilities of new technology can be implemented in the design of new products. But also we are finding inspiration in nature and implementing it into functionality of the product. But since I started teaching, I was assigned to teach subjects like eco-design and design with social impact, and I was reading more on this area, especially design with social impact, because it was pretty new for me as a mechanical engineer and designer. And I somehow stumbled upon some identical crisis, because I figured out that with the sustainability, we as designers just produce more products and more waste in some way. So I started working with the students, because I at the moment have a lot of subjects, and I teach a lot at the faculty, so I don't have a lot of time for research. So I treat my students as lab rats in some way, and I tried to find new approaches. So because this is pretty new for the Faculty of Mechanical Engineering, we are working with steel structures and things like that, and I wanted to implement something new and to shift a little bit the design approach, and actually to start thinking from the material, and to see what can we come up with that, and how can we rethink the design process and the product development. So we started experimenting a lot with materials. So as one of the reviewers of my paper said, that maybe probably this will be more adequate in the second track, but I thought it would be more appropriate here, since I'm concerned more about the application and not the material itself, because I'm not material scientist or biologist or chemist. So we were only experimenting with the materials. The idea was to gather materials that can be easily found in our area. So something that there is abundance in our nature, in the area that we live. So we consume a lot of coffee. We have a lot of nuts growing. So those were the materials that we start investigating. So we were at first we were only experimenting with grinding materials, with compositions, just to see by playing, learning by doing, to see what kind of composition we can make, what kind of structures. We were concerned more about the details that can be, not the composition of the material, but to see how they can be molded. Are there any cracks that occur? What happens afterwards when Tamara started investigating a little bit more for master thesis, we involved more people from technological side, and we were reading more about research papers on this topic. But idea here was to work as an to reinvent the whole process, to involve as many people as possible. So we were also very often doing workshops with high school students and inclusivity workshops with students with disabilities, where we were learning by play. So the idea was to experiment with the material and to come up with design lead by the material, in some sense that will familiarize people with these new materials. And from other prospect also, as the speakers before me were talking earlier, to familiarize, because there is a risk of people adopting this kind of new product that are made from waste, or that are biodegradable, that they are not valuable enough, and presumptions like that. So that was the main idea why we tried to involve also consumers and non-designers in this this whole process and this whole approach. And mainly we weren't so as I told, we were inspired by the material. So there wasn't a specific topic what to be designed at the first, but the idea for the products arise later when we understand better the materials. So these are some of the examples. We got bioplastic from glycerin, we got some children toys, and even furniture made from coffee grounds and nutshells and stuff like that. So the idea with this mine presentation was to show that there is application. This is actually at experimental stage at the moment, but we are working in this area, and we want to try to implement it on larger scale to make it even some pilot projects. That will be from my side. Thank you very much for that.
Thank you, Elena. Okay, so now we will have a presentation by Kailash Ramlal, if you want to join us, please, who is a laboratory manager and a specialist technical instructor at the Royal College of Art in the UK.
Hello, everyone. Okay, thank you very much for the introduction. Thank you to the organizers for having this project here. Let me move over to the slides. So before I get into the presentation properly, I'm just going to say that the sort of lead applicant on this project, Marcos Jojo, couldn't be here to attend the conference, so I'm hoping to deliver this in his stead. So yeah, this is a project that started during his time at the Royal College of Art in London on the MA Design Products program with his tutors Spiros Pofilatos and Ali Mohammed, and I've joined later on the project to kind of attend in a more technical support capacity. But the project began with a question of, can I grow a glove? And this project came about by the sort of design interest in bacterial cellulose as a material. So I think I don't need to be explaining too much what the promise is of biomaterials because of their inherent kind of circular or potentially regenerative characteristics, and the fact that a lot of biofabricated materials, when are produced by a biological system directly, often have kind of very excellent physical properties that are then need to be modified and adapted to the human material manufacturing kind of systems that we've developed. So the limits then of these current systems are actually compromising the radical potential that bio-based materials have because of the fact that they're forced through processes that they weren't designed for them, really. They were designed for mass manufacture, often subtractive manufacturing, and require kind of chemical alteration to then try and be paired up to the characteristics of synthetic materials or petrochemically derived materials. The material itself, as well, from a more material-focused perspective on bacterial cellulose, is that often production focuses on either two different streams, so producing flat sheets as it grows between the interface of the nutrient medium and the air, or in a fermentative vat or bioreactor, which produces kind of spherical cellulose that you then have to resuspend, and then you can do something with like spin into fibers, for example. What Marcos suggested is that actually there is a better way to try and work with this, and actually an advancement that we need to be able to realize the potential of these materials is infrastructural, is that actually biomaterials are not living up to their full potential because we simply don't have the infrastructure to use them in the way that they're going to give us the most benefit. And there are interesting conversations there around actually how we value that in the current kind of capitalist, consumerist financial structures that we exist in. But as a technical and technological solution to material manufacturing, Marcus proposed Hydra. Let's see if this will animate. If not, what we have here, Hydra, is a closed chamber machine. We use the word bioreactor a bit loosely, but essentially what we have is an axis along which we have a template or a mold, which then rotates and dips into a vat of nutrient broth. So in this case, if you're making SCOBY, you can just give it a bunch of very sweet black tea because SCOBY loves that. And what we found, or what Marcus found, is that we would get SCOBY growing in 3 dimensions along the surface of this entire template. And what we can do is you can actually take that material directly off of the mold and use it, and it has a 3-dimensional form. So as a technological solution, there are several different ways of manufacturing with Hydra that Marcus has developed, some of which are here. The one that we've been exploring most in the lab is external rotation molding. So this is just a single kind of mold that is then rotating. But there are other ways of biofabricating with Hydra that you could use for different applications. We've also found, because of the rotation, probably because of the increased oxygenation, SCOBY and bacterial cellulose that's growing in this way is growing up to 5 times faster than if you were growing it in a static sheet. We haven't got any comparisons with any bioreactor studies, and we haven't done that yet. But where we are right now is preparing for publication. We're just collecting some data on actually the application of the material itself and generating a proof of concept. So we'll actually go and finally grow that glove. And then what we're hoping to do after that is develop the machine further, develop the technology further, so that we can actually start putting it towards a few applications that we can describe and validate that the system works. And so I think one of the important parts that I wanted to leave this on is both the interdisciplinary nature of the project team itself, and this is a big schematic that Marcos came up with, but also the fact that the position here is that developing infrastructure for biofabrication and manufacturing using bio-based materials is a vehicle to help cohere between technological developments that will continue to hit the market and social and cultural practices of care and circularity. Because the practices that we use currently for material manufacturing simply do not fit and actively compromise the characteristics of the materials that we're trying to put into them. So we have Marcus on the project up there and his 2 tutors, Ali and Spiros. When this was first developed at the RCA, we also had Maria, who is a biodesigner and creative technologist that runs the RCA's Natural Matters Bio Workshop for its students. And then finally myself and my colleague Miriam from the Center for Materials Science and Culture joined at a later date. Myself, I run a research biomaterials lab at the RCA, and we're developing this work in there. The reactor is in there idle at the moment, but it'll be switched back on when I get back on Monday. And Miriam is advising on more of the distributed manufacturing aspects of the project and its potential. That's it from me.
Thank you. And now we welcome, of course, the person you know very well, Pierre Oskam, who is the vice chair of DESIGNAE, but he has also many other hats, right? Yeah. So like co-founder of Urban Reef, co-founder maybe of Regenerative Futures Lab. Yeah. Specialist asset manager, I should say, manager for architecture and sustainability in, what did you call your?
It's Stedin. Yeah.
Stedin, right. Thank you very much.
Yeah, thank you very much. So what I wanted to share today is actually, I work a lot in 3D printing with ceramics, making bioreceptive environments. My background is landscape architecture. So I'm also really thinking about scale. And 3D printing for me, it's very interesting to do those research and experiments on that level, but to go to this macro scale, I was thinking in a different way. Can we make something that is very local and doesn't really work with all those different technologies? So can we work with what we actually find on the location? So I was inspired by the tide, so also the landscape here around in Aveiro, you can see that it's actually shaped by the tides. And the Netherlands as well, this part in the Netherlands as well, created by 1.5 meters of level of water difference. So by shaping that, the whole culture there is shaped, also ecology. And it's a very rich environment. So this tidal pool here creates a lot of interesting biotopes. And what's actually happening in the cities, so this is in Rotterdam where I live, it's a harbor city, you see that those environments are erased because we want to control this environment, we want to control and see, it has to be functional for boats to go there, of course. So this richness and also this idea of regeneration, shaping land and all the life that's around that is lost. Here you can see in red are all the areas where you have this kind of quays. And the wish of Rotterdam is actually to become greener, to do things with green waterfronts. They're making some parks on the water sides, but they're not doing anything with the tides. Well, you have 1.5 meter of tides here, and I think it's a shame. And we also contacted some biologists and ecologists around. They gave us some tips and to see that that's indeed true, that there's a lot of interesting life happening. So the proposal was heavily inspired on work of art ecology, where they make, with community actually, they create these little tidal pools on the walls. And we thought as well to look into that idea, but make them a little bit larger and use the materials that we find in the harbor itself. So the proposal was to make 10 of those baskets and fill them with certain materials found in the area. So first we got together and we took roots from a reed. So it was actually found somewhere else in the Meuse. We went to this circular hub where they have different stones and dredge, as well as sheet pilings. So it's actually at the same location that we were working. We took away the sheets and we brought them to this place to make these different baskets. The dredge was actually, is very polluted. So we took this away and we went to this place where they cleaned the dredge. So using all those different materials, we made the project. So we installed 10 of those little baskets last week actually. And it was quite, they were like wielded against these quay walls. And here you can see on the right that with the tides, they will get filled. So you have these little tidal pools of 60 liters. And at this point, we just placed on different, we did experiments, so we have 4 different heights. Some of them have the reeds, some of them have the dredge and the stones, and some have nothing. But I was as well thinking, so for me it's like a little experiment. So it's installed last week. In 6 weeks, 6 months, we will see what's happening there. But I think as well it can be a platform for ideas. So I want to share this with you as well to say if you have any ideas for this, or how this could be a part of a research, because we're going to do it, at this point we're going to do the monitoring as well manually by just going there every 6 months, but this could also be optimized, or we could work with photography, or, so I'm just, for me this idea, this is just a little side project that can instigate something new, I think. So if you have any ideas, feel free. So yeah, that was me.
Thank you, Pierre. Jelena and Kailash, if you want to come, and we will do a bit of session of questions and answers. Okay, so we have actually a lot of questions. The first question for Jelena. Do you use co-design with the more than humans or just the people and like students? Do you use co-design with like also in the broader sense of like with more than humans or only with like people?
Yes, so the idea is to do it with people from different backgrounds. So mainly we, depending on the problem, mainly it's easiest to do it with students and also with faculty students, design students, combined with students from different backgrounds and high school students, but also pretty often when we have the opportunity, we do it from people from different backgrounds and from the industry, and we have a few examples when we were working with that's the main idea. This is just some modeling and shaping the methodology with the students, but actually the idea is to have it on a larger scale and to have it with students for professionals from different backgrounds like biologists, engineers, designers, sociologists, psychologists. So far, because of our connections and background in mechanical engineering, I have experience only with the industry and some NGOs on different smaller projects that been working.
Thank you. Question for Pierre. Are there any hope for the Venice Moss project to I guess I'm completing the question, to become fully nature-centric and biodesign?
For the what project, sorry?
Venice Moss. Moses? Mose? M-O-S-E? Mose?
Oh.
Yeah, so they have this whole project called MOSE that is some sort of, as far as I know, I'm not an engineer, that can predict and adapt in real time in case of a huge rise of the tides and the city becomes, so it's some sort of benchmark for every coastal area. So I was thinking if we can see this type of project, as you have this project, in a major size, you know, or not?
Yeah, so yeah, that was actually the intention also of the last slide, to thinking about this scaling up and scaling out. So in the one scaling out, so how would it look like if we do this in different places and also use the local materials to do this? So not saying that it should look the same way everywhere, also depending on of course, the quays, how they look like, but also the kind of environmental conditions of the ocean there. So in that sense, already imagine how it could look differently in different locations, and scaling up indeed. So that's the next step, is to think of, okay, with the monitoring, we will try now to find some indications of that it does something for biodiversity, and maybe as well indeed for water flow, like this Amazonian mangroves you were referring to, right? As nature-based solution for flooding. Yeah, so yeah, definitely, and to think of this even on a larger scale, how would you design quays in a different way? Not just little baskets, but maybe they should be underwater, different levels or something. Yeah, yeah, that's what I mean. So can we scale this into something else? Right now, we could also use this as a test bed to lay in different materials that we 3D print, for example, with very intrinsic ways of algorithms. So we have all the porosity that is needed, and at the same time reuse materials or biomaterials and test them. How do they behave to this tidal area? And I think the tides are very interesting because this is a force of nature that shaped the world, as it still does, and we should work with that. Yeah.
Thank you. We have a question for Kailash. Is the person here?
Yes.
Thank you. I'm just asking because I'm not sure to understand the question.
Yeah.
But please, could you?
Other question.
Yeah, but it's interesting.
Yeah. Can you? So yes, the bacterial cellulose question. So it is known from the 70s that the acetic acid bacteria, the Comagatae genus, has this fantastic instability, natural genetic instability, to find themselves in a position of less bacterial cellulose production. So in terms of anthropocentric way of using this particular bacteria to industrialize bacterial cellulose, it reaches some sort of impossibility. So what are your thoughts on this?
That's a very big question, I think. I think, well, I don't know what I think. I'll start there. I think the question surrounding, what we would all consider the ethical extraction of material from microbial systems is something that is being quite heavily explored from a design perspective rather than really from like a scientific or an industrial production perspective. I think this is perhaps one example that we have right now where there's a really tangible output from a relatively kind of low cost or low effort from a human perspective, a very low effort way of extracting material from a microbial process. Like you say, it's been known for, humans have produced SCOBY for thousands of years. We know this material extremely well. And there is a wealth of other microbial metabolic diversity that's out there that we maybe don't have a tap into just yet, which could provide interesting ways of working with these biological systems as opposed to continuing to try and engineer a system that, like you say, evolution has already decided that it has a limit, And I think that is going to be a really important transition for biotechnology, because I think a lot of biotechnological innovations right now, even Hydra, it is capitalizing on a biological function that we already know. Yeah. But I think something that is interesting about Hydra is that we would like to, at least in how we continue to develop it, develop it in such a way that it is adaptable to future discoveries, and that will decrease our reliance on certain kinds of biosynthetic pathways or certain kinds of organisms, and perhaps even decrease our reliance on having to heavily engineer microbial systems to work for us. And I think that's also one of the nice things about having a deployable is the word that Marcos would use, but like a distributable technological solution. You know, this is a machine that ideally is not going to live in a lab. There are going to be tens of these spread around a local ecosystem, and it can be accessed in a needs-based way, and it can be adapted to a specific use case that is then required. And there are some elements of biology that can then be worked as part of that. Does that answer the question? Sure. Yeah.
Question for you, Jelena. Have you been met with limitations on the potential uses and the kind of like prospects, maybe scalability of the designs, like based on the sources of the waste?
Yes, well, basically when we started thinking about the materials, the possible materials, we wanted to be something locally produced or locally abundant ways. So that's why we chose coffee, because in Macedonia there is a tradition in drinking coffee, so we have abundance. So, but also we were thinking about logistic, because and finding ways, and we designed some pathways how to produce it, actually how to secure it locally. But what we have as a limitation is the scalability of the product, and that is something that we are working, because we as designers only came up with shapes and forms that can be designed, and at the moment we are testing the mechanical properties. And surprisingly, the composite of coffee and ground nuts is extremely performs good on compression tests, but we don't have. I mean, we used only for the mechanical, for the chemical properties, only the researches that we found online. So we have to find resources and labs in Macedonia to investigate that part, because we have problems with molds and things like that occurring. And that's why I'm saying this is only not even a pilot phase, but it's really a prototyping phase of the whole process. But as we can see, there are nowadays products like this. So I believe that we can manage to come up with a product that can be manufactured on real real scale. Yeah.
Okay, thank you. We have maybe one last question that I think also Kailash could be also relevant if you want to answer. It was asked to Jelena, but I think maybe Pierre, if you have a take on this also. Do you? So more specifically about material productions, do you think that this kind of like new ways of producing could lead to an even increased consumerism?
I think when it comes to, let's use the example of bacterial cellulose and this bioreactor, the hydrobioreactor. The way that we are framing Hydra and this model of biomanufacturing is not just a way of producing material, but it's a way of considering a culture of material use. So what I would hope is that as new ways of making materials and using materials are developed, as technology develops, that cultures of sustainability and care are co-evolve with them. And ultimately, I think that is one of the sort of valorous kind of characteristics of these biomaterials, is that they require care in many ways to produce, but also to use, to maintain. It requires care in a circular system, because you have to make sure that you can give back and close the loop, or regenerate and make sure that your materials are accounted for. And I think it's that accounting for that then becomes accountability, which becomes care, which becomes a culture. So I hope that this way of thinking about manufacturing with biomaterials encourages people to also consider that the model of mass manufacturing, you know, has a lot of its own advantages, of course, and it feeds this model of consumerism, but there are still other ways to care about what you're using and what you can make if you have more of a personal stake in it. And so I think I think that there's the potential there, because I also don't think that consuming more necessarily tallies with this concept of biomaterial production, because it's slow. I mean, bacterial cellulose takes ages to grow. I mean, this is making it a little bit faster, but still, it's taking days or weeks, and you know, that I think is going to be the real way to turn these ideas just from lab projects or, you know, prototypes into actually becoming developed, embedded technologies. Is it requires culture around it. It requires that culture of care. Yeah.
I really like to, you know, when I work with mycelial materials, and very often people are like, what, it takes a month to produce, to grow? I'm like, how long does it take to produce the plastic you hold, right? But I mean, I think it, you know, for now also, it's also really interesting to you know, there's no hiding of the supply chain, of the transformation chain. It's in your hands, and that's also a transformative potential. Jelena?
The same question, right? Mm-hmm. Yes, I will have to agree, because actually I don't think that consumerism can be bigger than it's now. But also I think in this way we are not enhancing, but in some way we are probably making more sustainable, because the first is the care for the products, because you think you have something vulnerable. For example, a lot of these bioproducts that we are making need to be tended and care, like waxed with some, for example, if you have product from eggshells, you have to wax it with certain wax natural, so that it doesn't crack. So you have this tending and maintaining the product that gives some personality, and you're involved with the product. So we believe that then you will get maybe more attached to the product and maybe retain it and use it longer. But on the other hand, when you're finished with the product, you can put it in the compost and then in some way reuse it. So you don't create more waste if you dump it in the general trash. Yes, but if you again try to maintain it and handle it with care, then you can maybe some way not get a close circle, but very close to the circularity that we aim, instead of the linear process that we are using at the moment.
Yeah, so the same with the reason that I gave this presentation, not about 3D printing, because with the 3D printing you are dependent on this machine again. So I was thinking with this is maybe just an example of a method for working with the local materials, thinking about the local environment, and from there creating something. And it definitely is about care, caring. You have to go there, you have to give attention. So I think it's about a certain culture. So I would just think, like, for example, with the tidal pools, how in different environments would we make tidal pools? It's not about having one machine that can create them and place them everywhere in the world. It's about how people would relate to the river, how they would care for the tidal areas. So it's more that story, so going towards a regenerative culture. And I think that's where all this is leading to as well, that we need to have a different attitude towards everything around us.
Thank you very much.
One more thing on that. Sorry. Just, yeah, while you were speaking, it just reminded me, just the idea of caring, I think, is really important, that why we're interested, I think, in developing this kind of infrastructure, which moves away from a sort of centralized mass manufacturing perspective, you know, for biomaterials, etc. It's moving towards a system where your care has impact, I think, because right now it's very easy to feel, and perhaps even the data might show you, that you can spend as much time as you want caring for something and making sure that you put it in the right places that society or the system has lined up for you, but it doesn't actually end up where you think it does. It doesn't get recycled, or it doesn't go to where it needs to go. And so that system of accountability is broken. And I think the radical potential here is ensuring that that care, which is so important, has impact.
Thank you. That's a great note to end this panel, I think.
Thank you.
Thank you very much for your participation. And now Jelena again.
Yes. Well, we continue further with our next sub-panel, which is regenerative practices and social ecological systems. Our first speaker is Antonella Belletti from Politecnico di Milano. So I would give her the floor to get us knowing in the topic of life science knowledge infrastructure as unexplored resources for design. Thank you.
Thank you. Thank you for having me. So I'm Antonella Belletti. I'm a design engineer from Politecnico di Milano. I will present a research that is more, a bit more conceptual and contextual about how these kind of infrastructures can be used to generate and to share knowledge for the design practice. So this work explores mainly the intersection between design and the life sciences. And starting from especially biodesign and more than human practices that, as we know, are challenging the more anthropocentric and more than human perspective by promoting multi-species coexistence, ecological interdependencies, and regenerative practices. So as a result, design is increasingly being connected and is increasingly engaging with biology and in general life sciences more than merely inspirational practices, but actually seeing them as epistemical and methodological grounds. But despite this fact, many times this relation is often limited. In fact, many design practices that claim that to connect with biology and in general life sciences often use mediated and abstract interfaces. For example, using methodologies such as biomimicry or using generalized ecological ideas, data sets or laboratory protocols, which of course are used and are useful in the life science practice, but they dissociate a bit from how actual biological knowledge is created, which is through documentation, categorization, observation, and the management of uncertainties and practices of care. This detachment, I guess, is also a bit created by the misalignment in temporalities between these two different disciplines. Of course, design works at a much higher speed rather than biological knowledge, which is more cumulative and a slow process. So because of this relationship between these two disciplines, risk of remaining many times merely extractive, instrumental, and many times also purely speculative. So the problem is most of the time is that design does not lack interest in biology, but it lacks engagement with specific infrastructures that where biological knowledge is actually generated. For this reason, this work mainly aims to explore these kind of infrastructures and how these infrastructures like botanical gardens, national history museums, aquariums, or seed bank can be reframed, and how they can be reframed and understood by designers as long-established infrastructures dedicated to studying and observing and classifying life. So the problem is mainly that biology and the relationship between biology and design is missing a common ground, which is mainly caused by a difficult to access and also an incomprehension many times between these two disciplines. So the work, this study is mainly trying to reframe this kind of infrastructures as to create a shared common ground with design by addressing three main questions. So how can this infrastructure be made relevant to generate knowledge for designers in an effective way? So going beyond this, well, not simple, but going beyond the giving the merely biological information. How can a shared language be created between these disciplines, and how a translation can be given between these different disciplines, and how can this infrastructure become spaces of learning and collaboration with biodesign. So to do so, these infrastructures are being reframed as mainly relevant epistemic environments rather than simple repositories where samples and species are stored. They can be seen as a form of biocultural heritage, where knowledge emerges from the dynamic relationship between organisms, technologies, and practices of care. And how can they can be seen as boundary objects supporting the translations also across these disciplines. And with this, I conclude my presentation. Thank you for your attention.
Thank you, Antonella. I think DESIGNAE is also working on maybe solving some of the gaps that we heard from Antonella, but we will discuss that later. Now I would invite Cristiano Pedroso Rosado from Instituto Superior Técnico of the University of Lisbon to give us knowledge on biodesign solutions in fashion. So, please.
Yes, may I ask the colleague up there to turn down these three lights? That's okay? Okay. Yes. Yay, he can. He can. Yes. This presentation is a research project that I'm beginning in my free time, and it connects this connection between one of the most potent sociological system of expression, that is fashion, and biotechnology, design, biodesign, whatever the term they use, we use, people will use. pay attention. You see, it is just a clip from a longer video, and we have another one here. Está certo.
Every day we ask ourselves the same questions. What do I want to wear today? Who do I want to be? Do I dress for myself or for others? I know my choices will be seen, judged. But what about my skin? My skin is mine. It belongs to me alone. I decide how to present my body to the world. Shouldn't my clothes give something back? What if there was a new way to get dressed? A way that didn't just express who I am, but cared for me too. Clothes that care for my body as much as I do. I own my image, my body, my skin. I wear more than fashion. I wear the difference. Plus.
If we are in the age of the greenwashing, after more than 20 years knowing what is happening for the climate and choosing not to do the necessary to change, we are also entering into the biodesign washing, specifically in fashion. The point is, in the first case, Iris van Herpen, in her collection Sympoiesis, is doing this collaboration with the Imperial College to produce a capsule collection to show what can be done. However, what can be done is not enough to help us achieve a practice of care and to bridge the boundaries of our multidisciplinary fields in order to achieve some sort of co-design together with the more than humans for multi-species coexistence. My body is not only mine, it's mine, and it also belongs to the millions and trillions of bacteria that helps me to live, and I help them as well as one. However, fashion is doing the business as usual. They are producing, designing for consumption. And in my personal perspective, we as humans already did too much. The designers did too much. The engineers did too much. We need to produce some sort of step back and contemplating what is happening right now. And we heard the case of the bacterial cellulose. These the Comagatae they have a natural genetic instability to disrupt themselves And this is not being discussed enough in biology, and it has been appropriated by design, by designers, by fashion designers, to couple bacterial cellulose with petrochemicals and to make capsule collections with Gani, with Stella McCartney, and who knows more. And we should know how to choose the right bacteria, how to define the growth conditions for the desired output, how to know, how to learn, how to measure the genetic and epigenetic mechanisms that are required for industrial applications, but also for classroom designs. It's not a playground, guys. It is not. It's serious business. And as we just saw, the entire planet is looking at biotechnology, biodesign. People will follow. Zara just put the jacket on Bad Bunny Super Bowl concert. Why Zara? Why? Why? It's our job as bio designers, coming from a diverse array of siloed-based fields, to cross these fields and to say, the experts are here to help, but we need to contemplate and not be so eager to design. And so this is my final take. And as Bruno Munari said in the late 60s, creativity without rigor creates the illusion of art. And we are not artists. We are here to help, to guide, to mediate all these fields in a complex world where the players do not know, do not want to know, and they lack the power of will to change. Thank you.
Thank you, Pedro. Cristiano, for your interesting presentation. And now we move forward to the last panelist for before the coffee break, Nuria Goldáraz-Salamero from University of Zaragoza, Spain.
Thank you for this opportunity. So my background is in industrial design engineering, and in our research group in Zaragoza, we work in this line of research in which we try to use design for coming up with solutions for circular economy challenges. But it's also like a multidisciplinary kind of research, because I also work with the Agricultural, Forest and Food Science department, where I carried in the University of Turin, where I carried out my PhD. And today I wanted to bring to you a case study regarding the hazelnut sector. Okay, sorry. So to give a little bit of background, hazelnuts are one of the world top produced nuts. It had 1.2 million tons of production back in 2024. And this has a drawback because it also entails the generation of a significant amount of waste, because 55% more or less of the nut is actually discarded. And right now is used as for incineration for energy generation. And so what we want to do with this research is to come up with new methods or new ways of valorizing these types of wastes. So we based our methods or methodology in 4 stages. First, it was the material exploration. Then we followed up with the co-creative processes, with participatory processes. And then we followed up with the further development of the concepts. And we finally verified our outcomes with environmental analysis. So the preliminary step, which is the material characterization, was focused on finding the properties, physical and chemical properties for hazelnut skins and shells, which are the two main wastes produced in the hazelnut industry, and which we use literature and experimental data for obtaining these properties. And then we introduce our data into a Granta design software, which gave us like the analogous materials to understand which are the possible uses of these types of waste. In the figure in the right, you can see like the possible analogous materials for hazelnut shells. And this preliminary step was essential to developing later the co-creative processes, in which we gather different people, different participants from very different backgrounds. And we developed these workshops into 2 sessions. Like the first session was focused in the generation of concepts. And in this session, there were people from the waste management, environmental science, agri-food and material sciences backgrounds. And the results that we obtained using the forced relationships technique, which is a creative technique in which we randomly combined the properties of the hazelnut shells and hazelnut skins with possible industry applications, possible industries. We came up with these 4 concepts, which were a biochar filter for water filtering, a biodegradable container, a xylitol for diabetic consumers, and the introduction of this waste into animal feeding. The second session was held later, and it was basically for selecting the concept, evaluating the concept, also with a different set of participants from different disciplines. In this case, animal feeding, wood technology, design engineering, and agronomy and more. In this case, we employed a different creative technique to proceed with this evaluation, and we used the positive, negative, and interesting creative technique, in which the participants evaluated these 4 concepts based on 4 different metrics with novelty, usefulness, feasibility, and the circularity. So the final concept that was selected in this second session was the animal feeding, which held the most acclaim from the participants. And so we came with the evaluation or the verification, sustainability verification of this concept. For that, we used the life cycle assessment methodology, which, if you are not familiar with, is a systematic methodology and standardized for evaluating quantitatively the environmental performance of products or processes. In this case, we did 2 case studies mainly, one for livestock feeding and the other one for pet food. In the livestock feeding, we compared 2 different scenarios, like the introduction of hazelnut skins into, oh my God, I didn't realize that it's not showing the graph well, but it's okay. So what's supposed to be on the X axis, thank you, is the environmental indicators. I don't know why it's so in numbers, but it's okay. But basically, we just monitored different environmental indicators, evaluating the use of hazelnut skins into livestock feeding and their current use as energy. We found out that for most indicators, there was an environmental impact reduction, showing positive results for the environmental, the livestock feeding solution. And in the case of the pet food analysis, life cycle assessment, we came up with 3 different concepts of pet food. In this case, we were focused on developing concepts for dog food. And we had a first concept of dry food, then second one of dog chews, and the third one, it was a snack. Also comparing to conventional or commercially available solutions, we found out that there was also a significant reduction in environmental impacts, in this case, in the carbon footprint. And so what I want to sum up with this research is that there are different insights, right? For us, it was key to have a preliminary step of doing material characterization of the skins and cells for later on developing the participatory process. Then that despite the diverse expertise of the participants, like the sessions were deemed fruitful, and we were able to come up with sustainable solutions that were later validated by the environmental analysis. Then I want to also highlight that applying these design approaches for circular economy challenges can provide sustainable solutions for hazelnut byproducts, and that also that these methodologies were not only successful in mitigating environmental impacts, but also it fostered or it promoted valuable synergies between the animal feed and the hazelnut industries. And lastly, that this integrated approach, combining multidisciplinary research in biomaterial science, participatory processes, and environmental analysis, may set a framework to evaluating other kinds of agricultural byproducts. Thank you so much for your attention.
Thank you. Nuria, you can stay with us for questions. I would invite other speakers as well to join us. So thank you very much for your insights. I think we went from more broader approach suggested by Antonella to very concrete applications of biodesign solutions. So we can start with the question for Antonella. What kinds of institutional or educational formats could help build a shared language between designers and life science researchers?
Well, of course, prolonged residencies, also probably from the side of designers into these kind of structures to first of all see how biologists or in general life scientists practice and acquire knowledge of this kind of organisms and of life in general, in order to then apply them in an effective way in design, from multi-species ethnography on site, and not just reading about them and acquire knowledge from maybe published papers or from short-term interviews to experts, but actual hands-on practice and prolonged also studies within these fields. Yeah, probably. I don't know if I answered.
So you would say
questions.
something like more hybrid formats of
yes,
learning.
Yes, I guess so. Yes, to generate common knowledge, because as of today, probably designers just apply knowledge from, as I said, just maybe interviews or quick interviews or yes, reading papers from biology, of which actually of course the language is different from designers, because the designers do not have a background in this field, so it's probably more interpretative from the designer side to apply this kind of knowledge, while collaborating actually long term with this kind of figures and yes, would actually create a more effective and more knowledge-based.
Integrative.
Yes.
Integrative knowledge. Well, I guess this is still to be the future of, I would say, education in more and more hybrid fields around nature and biodesign. We have one more question for you, which was, did we cancel Neri Oxman foundational work on these fields, specifically on biofabrication? So if you can...
Of course not.
give us the insights. I don't know who asked the questions, but
I mean, I guess. Well, I can draw this answer from my background. I did specifically, I did a bioreceptive study for my master thesis on bioreceptive concrete, and I guess this is more. Well, of course, we in Politecnico we have recently opened a biolab in which we are approaching mycelium and biofabrication with biocellulose and mycelium, and of course. But of course, in these kind of laboratories, we do not have biologists because it's a designer-based practice. I guess this is just a more grounded educational approach in universities and in the everyday studies. Of course, we have this big and with big foundings also, biological laboratories, but in everyday studies and in everyday practice and in a more, I guess, less founded biolabs, these figures are missing. And also in everyday teaching of biofabrication and biodesign in general, these kind of foundations I guess are missing, and the most practical approach is just interviews and short-term collaborations with this kind of professional figures. So I guess that's more the approach.
Mm-hmm. And do you think you will be able to maybe at Politecnico actually, how would I say, establish these environments that you think are needed?
I hope so. For my thesis, I worked with lichenologists and bryologists, but of course, also in that case, it was not a founded residency in these kind of infrastructures. So it's also, to return a bit to the previous question, I guess it's to understand how biological knowledge is acquired more than applied, in order to be able, as a designer, to not apply it on a single project, but just as a general knowledge basis.
Okay, and also from my side a note, I think that DESIGNAE can help exactly with these kind of environments, as it does seek to unify the terminology and obviously to connect all of us that are from very different backgrounds. So I guess this is exactly the place where we can make some advancements in the field. So I hope we will have good collaboration with Politecnico. Okay, so we can move towards the Cristiano. Very interesting presentation, thank you for that. And question for you first would be, first, the note that it is a good presentation. Isn't design doing its job by proposing alternative futures and triggering questions about which futures we want or don't want?
The two phases of design. So inside the lab, we do that. Outside of the lab, we produce chairs, biased systems. And anyone can easily see how many non-white people we have in the audience. So there is something wrong, and something wrong is the system. So as designers per se, we are doing both sides. We are creating the problem in order to solve it. So it's a bit complex.
Okay. How to stop people doing something if they do know how to do it, and then at the same time it results in ethical problems or disasters?
Yes, lack of knowledge is an issue for those who do not have the knowledge, but we have a globalized, connected information sharing community. So lack of knowledge is not an issue anymore. Lack of participation is. And we should invite everyone, everywhere, whatever the conditions, the social conditions, to participate. Do not close doors. Do not close doors. Open doors. Public organizations, public museums, well, we are closing doors for ages. And in this country, we are a still imperialistic country with very, very systemic social issues that compromise participation and compromise multi-species the concept of more than humans is, as far as my research goes, the biological beings, the built environment, all the social dynamics and cosmologies and beliefs. And we are not seeing more than humans in the participatory process. We are informed, inspired by nature and by people, and use them as resources. And we shouldn't.
Okay, thank you, Cristiano. One more question for you, although a provocative one, asking why the nature-based design is still sexist. I don't know who asked this question, but maybe we can discuss, yes?
It's more that, to show the image of beautified women during the scientific presentation is very sexist.
Yes, it is.
It's really not
It is not, it is not good. We should call out Copernic for doing this globally. They are instrumentalized. Well, all the hype culture, the influencing culture, are instrumentalizing the youth for feeding the same corrupt system we have now. And I mean, my job as a public funded researcher is to ask independent questions where they hurt. And I do, and I show. We have to show this, not to have the glamour of fashion, but to ask the provocative question. Why a black woman now appears almost naked? Naked. It's unbelievable. It's unbelievable. How the God are we doing in here? The boards of these companies, they have all the resources, all the skills, all the time, all the money, and they are doing this. Something is wrong, but that's why we have cost actions, European funding, to be creative, autonomous, and independent, to perform provocative research and to communicate and widely all of this.
Thank you, Cristiano. Because of time limitations, we have to move forward, although the discussion is interesting. For Nuria, how did you select the concepts? Were there others you discarded in session 1 or not?
Yes, there were others that, because the research was carried out quite a long ago, I don't quite remember specifically the other concepts, but in the first session it was actually the participants which, after coming up with the first ideation stage, they gathered together and discussed the ideas to come up with these 4 more defined concepts. Yes.
Another question, what do you see as the main barriers to scaling these circular solutions into real industry practice?
I guess it depends. On one side, of course, is the willingness of companies to scale up these researches. On the other hand is the lack of some background or knowledge. In the case of specifically the feed industry, there are restrictions regarding the actual application, just for safety reasons, right? And there are policies that are actually in a way hindering maybe some of these solutions. It depends case by case, and I guess it depends on specifically the different circular economy applications. But yeah, there are many challenges regarding this, yes.
And then last question to all. Last question to all would be, which kind of let's say results would you be satisfied in, let's say, next few years? What would you like to achieve, respectively regarding your own fields and current research teams and engagements?
Well, I think in a way, as we are in our research group, we are mostly designers. And as Carmen said in this morning, we are very horizontal. Sometimes and I think this DESIGNAE cost action is going to kind of help us with this, is that we need to be more concrete and have more knowledge regarding biodesign practices and natural sciences solutions. So I don't know about what outcomes specifically we are going to come up with, but I hope we can build synergies with different people from these specific fields to carry out forward more collaboration between designers and natural scientists.
Okay, thank you.
Thank you. So as I'm part of the material design for transition group in Politecnico di Milano, and as I mentioned before, we recently opened a biolab where we are trying to experiment in many different fields. All of these are mainly material driven. We are very multidisciplinary group, all of which are designers, but from different fields. So to increase actually even more cross-disciplinary interaction, as I said, would be great also to grow and to develop research further. So with yeah.
Mm-hmm. Less.
Thank you, thank you. I would thank to all of our panelists, and we can now proceed to a coffee break. Yes.
Thank you so much for the panelists. Thank you, Yelena. Thank you, Adrian, for leading this. We will have 2 photo shoots now. One will be here, okay? And the other one will be outside. Gonçalo? The other one. Gonçalo? So we will, I'm going to ask every member to come forward. And so we will take the picture here and the other one on side. And then coffee break. Where is everyone? Are they still in the coffee break, perhaps? Okay. Gonçalo, vais lá e dizer, tipo, venham para cá. E diz ao Arthur para ir falar com o coffee break. Okay, I'm just going to introduce the 5th panel. Wait. Ah.
It's me. It was me.
Okay, wait. I need to reboot this. Okay. Unfortunately, we cannot have it full screen. Otherwise, sorry, it was me. No worries. No worries. Ok, we have it here. Opções de exibição, tela inteira. Iniciar a apresentação. Lionel, estamos aqui com um problema de ecrã. O que é que se passa? Okay.
And then I sit down.
Okay. So Gemma, I'll put this.
Yeah.
Okay.
Okay. Okay. Thank you.
There you go.
Okay. Many thanks.
Good?
Many
All right.
Amazing.
Okay.
Thank you.
Hi. So hello everyone, welcome back. We are close to the finish, I promise, but this panel is very important, so stick with us. So this panel is for regulation, law and policies regarding biodesign. With us we have Professor Gemma Andrion, Mr. Patrick Schenker from Dark Matter Laboratories. We also have two invited speakers, which I will leave Gemma to present. So.
Thank you so much, everybody. I'm confident that you all are wide awake after a coffee break. So I give the floor immediately to our leader of the working group 5.
Okay, leader is a bit much. Hello, I know it's been a really long day. Our panel might feel a little different from what we've done today. We've talked a lot about biodesign at large, materials, fabrication processes, but in the spirit of making this truly multidisciplinary, we're going to look into some of the broader legal, policy, and sort of regulatory issues that exist around some of these domains. We're going to be focusing on marine genetic resources as an example, in a sense, to get into some of these questions, and we have some very lovely experts to get into it. But Gemma is going to set the scene for us.
Thank you so much, Pratik. And we immediately jump into my presentation just to give you a general framework of, and already Pratik told us that this is an example. We wanted to start our path for the next years with this case that is really a living case study because we, the BBNJ agreement, Biodiversity Beyond National Jurisdiction, this is the acronym, is in force since this January, so offers the opportunity to examine the intersection of international laws, science, governance. So we have this, and here in the picture you can see what are the spaces of ICs, so beyond national jurisdiction, the marine space that is located beyond national jurisdiction. So the main goal is actually to introduce some general research question that Working Group 5 will explore over the next 2 years. But just some key facts concerning this agreement that has been negotiated since 2018 2015, and now we have actually the picture is not updated, but we have 83 ratifications so far. So it's already a big success, considered that is the only multilateral agreement agreed and also entered into force of the last years, as you are well aware of the international situation and also of the crisis of multilateralism. So this is an agreement that is also considered as 4 agreements in one, because is based on 4 pillars for chapter. One is devoted to marine genetic resources and benefit sharing. The other one on area-based management tools, so the possibility to create marine protected areas or area-based management tool in on the high seas, so beyond national jurisdiction, where at the moment there is the principle of the freedom of uses of that space. Then part 4 is environmental impact assessment, and part 5 capacity building and marine technology transfer. So in those days in New York at the United Nations seat, there are the other negotiations for the PrepCom for regulating the Conference of Parties functioning, so that will be the international decision-making body of the agreement, and also the other technical committee, as, for example, clearing house mechanism that is very relevant for marine genetic resources. One, just to highlight some foundational issue that one is that we are dealing in our working group with legal aspects, so in this case international law, but also ethics. So we really need a clear distinction, because international law is a regulatory choice made at a specific historical moment by states, governments actually, under political pressures. And so ethics is something different, very different, because can offer orientation when law is absent, or when so ethics is not legally binding. This difference is mainly important for us. But just another reference, general reference to who speaks international law, who are the subjects of international law. Mainly, you know, are states intended as governments, not people, and international organization. But in the last 40 years, I would say, this circle of subjects is expanding. So we have also individuals that are considered in a way as quasi subjects of international human rights law, companies also, and NGOs and indigenous and local communities, as we will see, but actually they are still right holders. Now, another important, according to me, subject is the common heritage of humankind. That is recognized by the UNCLOS Convention, that is the United Nations Convention for the Law of the Sea of 1982, for the first time in the history, for abiotic resources only. So for the abiotic resources of the high seas, we have this principle that implies no non-appropriation, the interdiction to appropriate those resources by states in an autonomous way, and the possibility to exploit those resources through an international system, that is the International Seabed Authority. Here we have also benefit sharing, that is very important in and the developing states are beneficial of benefit sharing of the exploitation from developed states. But in the negotiations for the marine genetic resources of the ICs, this concept was very highly discussed, but because there was no agreement among states on the application of this concept. Thank you so much. So the solution was to introduce the idea of benefit sharing for those resources. Very quickly, I already said that this is a regime in construction, so all these aspects will be considered and regulated by the COP when the COP will be active, and this governance architecture is very interesting for DESIGNAE as a live design process. But I immediately now come to other two aspects that will be discussed by our panelists today. That one is traditional knowledge of indigenous people, that is the relationship between scientific knowledge and traditional knowledge has been widely discussed during negotiations at BBNJ, and because of this deep tension between Western scientific knowledge production versus indigenous epistemologies. So we will hear soon after Alessandra Passarello, we will hear Danilo Garcia talking about this, but I give immediately the floor to Alessandra Passarello for an historical and socioeconomic reconstruction of the dimension of deep sea exploration and knowledge. Thank you so much.
Good morning, everyone. I hope this historical reconstruction will keep you awake. So I will tell you from the very beginning, I am very happy to be here. I am an historian of science, historian and philosopher of science, interested mainly in the evolution of marine sciences and the way these sciences can contribute to shaping cultural, environmental, political, and socioeconomic representation of the ocean. And of course, you might imagine that as an historian of science, my sources generally date back long enough to watch them with the necessary interpretative detachment. So of course, January 2026 doesn't really fall into my century of expertise. So what am I doing here? Actually, I believe, and I hope you believe it too, that sometimes an historical reconstruction on current affairs, either in politics, in science, or maybe also in international law, can help us reveal what is behind our current rationality. So this is where history somehow matters to contemporary science, politics, international law, and so on. So let's start from the very beginning. On January 17, 2026, the agreement on the conservation and sustainable use of marine biological diversity in areas beyond national jurisdiction too much time, not so much time, so BBNJ agreement, came into force. And as Gemma was saying it before, this agreement is an international treaty expanding on a previous agreement, the United Nations Convention of the Law of the Sea, the UNCLOS. And particularly it expands on two different matters. First of all, it extends the UNCLOS in areas beyond national jurisdiction, high sea treaty, international waters. And the second thing, it expands the UNCLOS with reference to a very specific kind of activity that in 1982, the year the UNCLOS was adopted, was not still very developed. We might say it was in an emerging phase. So what is this activity is the research and harvesting of a very particular kind of biological macromolecules. I don't need to say more about this because we have many biologists, so nucleic acids, and on the use of a particular kind of information, which is the composition in terms of sequence of nucleotides of these kind of macromolecules, what the BBNJ agreements refers to as digital sequence information. Now, we might be tempted to think that this kind of digital sequence information are just data, abstract data. What do we do with this data? But actually, scientists are using this data to study life in extreme environments. Here you see, for example, the discovery of hydrothermal vents in 1977. This is kind of environment that we didn't even imagine could be inhabited a few decades ago. But this is not all. Of course, these data are very important for science, for marine ecology, but advances in genomics and in biotechnologies are making this data more and more important for commercial purposes. What is possible today is to use this digital sequence information to engineer bacteria in order to engineer marine enzymes or other kind of proteins which do have a huge commercial value. So while we say that bioprospecting, the activity of biodiversity prospecting is scientific, it is also heavily, how can I say, embedded in a socio-economic context. Only in the last 20 years, as you can see, there are many different oceanographic campaigns, such as, for example, just make yes with your head. If you heard about the Tara expedition, the global ocean sampling, the Malaspina expedition, all these oceanographic campaigns basically aimed at funding marine genetic resources and using digital sequence information to know more about the oceans and, of course, to exploit this digital sequence information in a socioeconomic context. As I told you, bioprospecting is not just about science, it is embedded in a geopolitical context marked by huge inequalities between countries who have different kinds of developments concerning technology and scientific expertise. And this is why some people are recently advocating that bioprospecting, if unregulated, could turn into biopiracy. And of course, this is why we do have the BBNJ agreement, and particularly the part 2 of the agreement, dedicated to the fair and equitable sharing of these benefits. So let's see. If we go deep into the text of the agreement, we can see that access, physical access to this kind of samples is very finely regulated. But what happens with access to digital sequence information? This is only loosely defined. And we might not be surprised because, of course, you know, digital data is something very intangible. So sometimes we know about scientific data, but it's very hard to imagine what scientists concretely do with this data, and also, consequently, it's very hard to imagine how to regulate this access. And this is where I think history of science might come into our help by giving us a different perspective. So this is the question of the day. When did we start to consider scientific data of the ocean depths as a source of value? And when did we begin to regulate access to this data. Now, I'm not going to go for a poll because we do not have so much time, but I will tell you that actually oceanographic research has not very ancient roots. The first oceanographic expedition date back to the very late 19th century. Probably some of you heard of the Challenger expedition. I'm sorry, I should have brought some water. Anyway, so don't worry. Oh, thank you very much. So the Challenger expedition was the very, very first scientific oceanographic research taking place in 1872, from 1872 to 1876. Thank you, Pratik. And we, in history of science, we generally think that this expedition is a purely scientific expedition. We mark it as a milestone of oceanography. But we know today, thanks to other historians who wrote it, that national prestige, imperial ambitions, and sometimes also economic interest were motivating these and other expeditions. If you see, for example, this picture, you can see a map of telegraphed cables. Actually, mapping the ocean was a way to get information on the seabed composition, on the composition of the sediments, on the water temperatures. And all this information were instrumental to engineers in order to plan where to lie cables on the ocean floor and maybe to predict the efficiency of the signal transmission based on the temperatures. So we can say that from the very beginning, oceanographic research and deep sea scientific data were capable of generating value, and there was no international framework regulating access to this data. What we had is only political, how can I say, political compromise among nations. The boundary between openness and secrecy was politically negotiated. Now it's time to... Here we are, much better. So what I was saying is that the boundary between openness and secrecy was politically negotiated, but not legally constrained. Now I will move forward to another very important period in the history of science, which is the Cold War. You know that, for example, the ocean were very important for military applications. There were many scientists at the time who were studying the permanent thermocline. Your scientists, you probably know better than I do what is the permanent thermocline, but if you don't, it's very easy. It's just the layer of deep ocean water when we have a sudden decrease in ocean temperature, and this decrease is capable of affecting sonar propagation, so acoustic wave propagation, and so to affect also military strategies, military instruments for underwater surveillance. So what you can see here is that actually already in the Cold War, oceanographic research had strong military applications, and these kind of military applications were considered to be instrumental, were actually at the basis of oceanographic expedition. Now, I think I'm going a little bit not too fast, so I will move forward to 2 slides. Here we are. So I don't know if you've heard ever about the International Geophysical Year. This is an experience. It is actually the first international post-war cooperation initiative during the Cold War. And this is the very first time when we have data about the ocean shared between all nations, even if these nations were not belonging, let's say, to the same part of the Iron Curtain. This kind of example in the history of science can help us understand a little bit how the circulation of data, even if we do not have a legal treaty binding states to share the data, can happen because of practices in science. So for example, if you go into the archives and look at the papers from the International Geophysical Year, what we can see is that scientists and the designers of this initiative had organized a very, very controlled system, a distributed system of world data center, physical infrastructures where data could be stored. Scientists, so imagine you are a scientist, you have your data taken from the ocean, temperature, salinity, and so on. You send to the nearest world data center, they collect your data, they file your data, they process your data, and then the data are there for whoever you want, whoever wants to access them. So for them, in 1957, during the International Geophysical Year, this kind of mechanism was a way to assure openness, open access to data. And despite this, what we see is that actually the World Data Center, even if they had to be a mechanism to make the data open and accessible for everyone, they were geo-localized and reflected the geopolitical tensions of the time. So you can see that World Data Center A is in the US, World Data Center B is in the Soviet Union, and then you have World Data Center C in Western Union and Japan. So what I want to say is that even if we have international scientific cooperation, even if we have a rhetoric of openness of data, we still have some mechanism to control how data flow. And here we come to the very last part of my presentation, which is today science. Today sciences are very different from the sciences, the marine sciences of the 19th century and of course of the Cold War. What changes in data circulation is first of all the speed and also the amount of data production. So what we see today is that digital sequence information which are produced through the extraction of marine genetic resources are an important and are actually fundamental for marine biotechnologies. In some fields of marine biotechnologies, such as, for example, in Mitta genomics, scientists no longer need to isolate bacteria and to cultivate bacteria in the laboratory because they work with digital sequence information. So we need to regulate access to those kind of materials, and we need that access to be open and at the same time to be controlled. So what I want to say here, for example, you see this is an example of a digital sequence information taken from the Tara expedition, and you can see this is basically raw data. These are easily accessible, publicly accessible from a public database. Everyone can take it, but the problem is which kind of data, where they are deposited, and most of all, who can have access to them. What you can see here is the International Nucleotide Sequence Database collaboration. This is a collaboration between 3 genomic databases. The first is GeneBank from the US, the second is DDBJ from Japan, and the third is INA from the European Union. The basic thing is the same thing as the International Geophysical Year. Somehow you have 3 databases from 3 different places in the world, and they all exchange information, so they are supposed to have the same kind of data and to make them accessible to every researchers in the world. But which kind of data do they host? Basically published data, so not data which still have to be processed and still have to be published, and raw data. So they don't publish, for example, accessory information on the environmental samples. They don't publish, for example, they publish raw sequences but not annotated sequences. This really depends. This is up to the researcher, what he or she wants to make public. And this is where the BBNJ agreement is trying to make a difference. Actually, once you go, imagine you are a scientist, well, you are not a scientist, you are a company, and you go in areas beyond national jurisdiction, you take samples, you extract information from the samples, where this kind of information should be deposited, and when, and who should be able to access this information. These are of course, those kind of questions an historian of science cannot, of course, solve, but these are the kind of question I think, and here I conclude, that a historian of science or a philosopher of science can help to resolve by bridging the gap between the language of legal researchers and the language of scientists. And actually the main point is to make more concrete, to bridge the gap between the formal language of policy and the material practices of science. And here I conclude. Thank you very much.
Okay, thank you, Alessandra, for helping contextualize a bit of what we've been talking today in history and in culture. And to kind of extend that argument, I'm going to invite our second panelist, Danilo Garcia.
Thank you very much. Hello, everyone. I would like to start to thank Nina and all your team for the kindly invitation, and also to thank my colleagues, Alessandra and Gemma, that introduction permit me to start in the very, very important part that we need to know. In the international law and in the legal part of the protection of marine resources, not only the genetical marine resources, but all of marine resources, we need to understand two things. First of all, that the states, we have one principle that sovereignty is the principal thing to governance that resources. But against this principle, we have all economics and political issues that not only the private company, but the same public company of richest states or developing states needs also to have access. Through all the day, I hear a lot of wonderful interventions about how we can to develop our researches to the clothes, to the health, for everything. But who is the proprietary of ocean? Who is the proprietary of marine resources? And in that question, we have another two problems. The first, that who make the ruler? Who make the ruler has the power. But how we can make the ruler with the data information? And the data information permit us to have all the control, not around the world, the states itself. And in the timeline of evolution in international law, the different states will try to regulate the access, the use, and the benefits. We started with the Convention International Trade Engaging Space with Fauna and Flora, the named CITES, and the United Nations, the Law of the Sea, the UNCLOS, in 1982. But that was not a completely agreement. Actually, we name in the international point of view, we name it as a constitution of oceans, but it's not completely, because that was only a good agreement to make the different borders, boundary, and the liberty that the states have to the ocean as navigation or fishing, etc. But the humanity, we needed a little more to regulate the access to the resources by the Convention of Tribal Populations or Tribal People, Indigenous People, 169, but that was also a good effort, a good thing, but not completely. And at the end of the last century, we understood that we needed to arrive to construct an agreement that permit us with the force, with the, not only with the convention or with our good volunty that permit us to regulate the use, access, and benefits. And it's like that who started the BBNG agreement in this year entered the force. But, okay. Now, theoretically, we have the agreement, we have the structure, legal framework that permit all states to regulate inside her jurisdiction, their jurisdiction, and outside, beyond their jurisdiction. But not only like that, that is in theoretical part. What is interesting in all that, for the first time, we included the indigenous people point of view, the indigenous people participation to take the decisions, and also to make the negotiation with the private companies. That is an interesting thing, but that was not enough. The different parts, the 4 parts of the BBNG agreement permit us to understand how important it is to regulate that part. The protect ocean or the make the different principle permit the legal world understood the different things. The first one, that the sovereignty of states is against the economical companies' necessities. And that is the big problem, because the states with the sovereignty principle wants also to have the control of to use, to access, and to benefit. But what about the difference between the rich countries and the developing countries? What is the difference between the biggest countries, not only in economical, but in extension of or the military power of each state? What is the problem in that? Because in the UNCLOS point of view, from the UNCLOS point of view, we need to start with the similar conditions. And the sovereignty of states governs now against all of these other points of view. The other one, that was the Article 7, this permit the general principles and approach. In this part, we have a big discussion. We studied that several months about the precautory principle. The precautory principle in this agreement, in BBNG agreement, is taken not only as principle in international law, but also as an approach. So we have another discussion in legal part. For us, for the lawyers, it's the biggest problem to understand that we can use only this part as a principle or as an approach, because of that depends all of mechanisms that we need to control several, not only companies, persons, but all the states couldn't to use or maluse or don't use the different marine genetical resources. And the effects could be very dramatic, obviously. Another problem, that was a big problem, but another problem that was obviously who can to have the access to that kind of information, genetical information, who come to have this access. And through 3 years
almost 4, the different states negotiate for to permit all the states in equal conditions to have this access. For our scientific world, that shows a good opportunity, but it's not open bar. It's not the open to make that we want. This agreement is also a big forecast for to regulate the propose. The propose that agreement make it in different sequences. The first one, the digital sequences information, that was not a practice under the notification, transparency, and benefits sharing mechanism. So that sounds like very weird, but no. The digital sequences information and how we can manage that in the practice, how we can to notify who can make it, needs another regulation. And that is the biggest problem, the almost last problem, because since 1982, the world think that UNCLOS, that was the best convention. That was a good convention, but not the best one, because we needed to have the regulation for the areas beyond national jurisdiction. Now we have that BBG agreement, but several months later, we show and we know that we need another agreement for to regulate that access. And what about the indigenous people? The indigenous people, from their point of view, they have the internal and intrinsic spiritual connection with every part of world. The concept of Pachamama, or the concept as Gaia, or the concept as one unity, a spiritual unity, we are part of them. We need to understand that the human is not the center of world and not the center of this, not only planet, but this existence, anthropocentric point of view. But we need to understand from their point of view that we can accelerate that conservation or that resilience for to recuperate, for to have another for another opportunity to have this kind of earth. Or we have the other, in the other hand, the faculty to destroy it all. The last one in this part. I would like to finish for the two biggest last problem of this agreement. That is the activities with respect the marine genetic resources. What kind of activities? The activity of collection, utilization, and access. If you don't have the technology to access to this information, it's very difficult to use and to share it. But this agreement permits the transfer of technology among every part. And the last one is the Article 13, the traditional knowledge of indigenous people and the local communities as the principal part to have the good conservation, not only to protect it, but to conserve and to manipulate, to share the technologies, and also to share the benefits, economical benefits, actually. That part is a little window that is open, but is not enough because, as I told you, is the difficult to make the line until what, or the limit until what we can to make as a company or what we want to make as an state. So that's the different challenges that now the international law needs to make the ruler and to make also the limit. But it's not only the state challenge, it's our challenge because our as scientific group, our as the population, our as the states, we need to participate in the same context, in the same line, in the same level. So I invite you to reinforce that initiative to make the compensation, not only the compensation to the indigenous people or the states or the companies, but the compensation to the humanity, because all of us, we can destroy the planet, or all of us, we can use the scientific things to prepare our new rule to the conservation and the protection of marine genetical resources. Thank you very much.
Thank you, Danilo. I don't think we have time for questions, unfortunately, but we will be unpacking all of these questions with Danilo, with Alessandra, with Gemma tomorrow at our Working Group 5 session. I'm a designer myself, and I think my takeaway from all of this and my invitation to everybody is to, first of all, I mean, history itself is a site for design research, is a place for us to begin our inquiry. As a designer myself, I've been guilty of this, of just making something in the abstract because it's, sexy or glamorous. But these things are rooted in a continuity, whether it is historical, cultural, legal, regulatory, et cetera, and those are things for us to consider. The second learning I'd like to, encourage the group to consider is just that the infrastructure we have, the legal infrastructure, internationally or locally, the sort of economic infrastructure that exists because of it, all of these are also sites of design. And a lot of what we make are informed and will inform all of these infrastructures. And it's really important for us as a DESIGNAE over the next 4 years to consider how our different pieces fit into this larger puzzle, and how do we not go about, causing more harm in the way that Christian described and so many other people described, and this biopiracy is a lovely word that I'm going to take home today, and do this right. So I'm going to stop my political speech there, and thank you very much.
Thank you very much, Pratik. Thank you, Gemma. Thank you, Alessandra, and thank you, Danilo, for this really interesting panel. Last but not least, the last panel has arrived. So I'm gonna invite Professor Antonella to please come forth. Professor Antonella is leader of, and Gonçalo.
Yes, of course.
Yes?
We agree that I will do the introduction because I have to leave.
Okay, I will stay.
Okay, thank you, Nina. So, we arrived to the last panel. I hope it will be a great finish. So, we're both responsible for this moderation, but unfortunately I have to go soon. But I just wanted to say goodbye. See you tomorrow. And say sorry for not being able to stay until the end, also because I'm co-responsible for one of the presentations, but I'll be here for more 10 minutes.
Thank you, Gonçalo, for sharing with me this initial part of the session. So I would like just to briefly introduce this last session that, in my opinion, is very important because we saw during those days that we are able to produce strategies, data, results, ideas, but then we have to communicate with the scientific community, general public, schools, and so on. Some concepts were already discussed during the panel today. Someone underlined the need to have a common language. Sometimes other people underline the need to communicate effectively among different disciplines when are under the same umbrella, like in our case, we are into the regenerative biodesign. That is our big umbrella, big boat, but we have different disciplines. We have to understand each other to be synergic into the field. So I would like to move to our first speaker. As mentioned, we have a fourth speaker today. I would like to invite to the stage Taryn Med, and she's going to talk about she's from the IE University, presenting It Smells So Bad in Here, Overcoming Design Student Aversion to Sensorial Experience in Biodesign. So how to train students to approach problem into the lab, maybe this will be your message. Please, the stage is for you.
Thank you. I think I need that, yeah, thank you. Thank you. Hi, thank you everyone for sticking with us here. Yes, so just a little bit about why I started asking this question. So I've been teaching sustainability to a range of professionals for many years now, 20. and through time, I've definitely seen my students change quite a lot over those years. And this last couple of years, I've been teaching Intro to Design Studio 1 in a Bachelor in Design program, and introducing biomaterials, right? So through time, I've seen the digitization of design and the corporatization of design and all of these trends that we've seen. With my colleague and I, Steph Fusani, who's online, hi Steph, we taught some students in Intro to Design Studio 1 about biomaterials. So in our curriculum, we were really, really digitally design focused in our first year initially, and we decided that this year to experiment a little bit and save like the digital interface for second semester in our core design curriculum. And so in restructuring the curriculum, we started to introduce biomaterials and these really sort of low-tech ways and ideas around material-driven design. And I should set the context as well that IE University is an international university, so in a classroom of 50 students, I might have 35 different nationalities, and our students speak an average of 3 languages, so it's a very, very diverse place, but also a very urban student body, I would have to say. So Steph and I are introducing, and some other faculty as well, we had a couple sections, we're introducing this concept of biodesign, and one of the immediate responses we get is, what is that smell? Right? And I kept hearing this again and again, and I had some students that were like, they couldn't tolerate this very sensorial experience that they were having. We live in a highly homogenized world. The majority of objects we interact with are human created. And it raised all these questions around, like, if we're going to be able to reconnect humans with natural systems and use biodesign as a process for doing that, how are we going to overcome this otherness that exists between us and mold, for instance, right? Like you were saying earlier, like I am 90% other organisms, right? 10% human cells. So how can we, with these highly digitized students and highly digitized environments and teaching environments, how can we actually approach the process of teaching biodesign and trying to keep students a little bit closer to the physicality of the world? So I will say also that this is in an early phase, and I'll have a little call for participation at the end. I've taught over 200 students now in some aspect of biodesign. So I sent out the survey to everyone that I've taught. I got 9 responses. I'm like, please, this is not an Amazon review. Please, like, let me know your experience. But it's really difficult to get their attention, maybe you've noticed. And then I had 2 volunteers for student interviews, and I'll be following up on those later. But faculty were very excited to talk about this. I had 5 interviews within 2 weeks, you know? So I'll have a call for your participation there as well, just to give you a sense of the sample size. So I have a degree in biology, but I'm a qualitative researcher predominantly now. So just to give you a little bit of the sense of the kind of data that we're working with. Okay, so I gave you a sense of some of the sort of context, right? So we, in working with these students, many of them, this is their first semester of design. They haven't done a lot of work in this space before, right? So some of them, their feelings were a little bit like, I'm not so sure about this. They didn't really know what to expect. Some of them really found it challenging to work in this space that is full of failure, but a lot of them really enjoyed that, and some of them found it too frustrating to want to do it again, right? They talked a lot about the messiness and the lack of control, as you can see there, being one of the hardest things to navigate, right? I think we're also training a generation of students that are very used to having a correct answer, and so this can be a really difficult transition. I won't go through all the data here, but in general, we introduced the idea of material-driven design, and many of them had never really tried to make a distinction between material-driven design and digital design, and so part of this exploration is to try to see if material-driven design, understand how they perceive it. So, like I said, a lot of data here. The kind of what's coming through, though, is this sense that there's a messiness and an unpredictability, and the sensorial experience of the materials made me uncomfortable, but I kind of liked it, yeah? So I think that there are some students who will never like this, right? And I think that's perfectly fine. I'll talk about this in just a minute, but in talking to other faculty who have taught this, weren't very many that were teaching this in mandatory classes, which I thought was interesting. There was only one other faculty member that introduced it in required courses versus making them electives, right? So I think there's a distinction there, and whether students can opt into this experience or if they feel forced. I think that's something we have to be really aware of, because this sensorial experience is not for everyone, honestly. And I think in some ways we might lose people and lose the audience a little bit if we push them into something they're really not comfortable with. But for the people that enjoyed it, they really enjoyed that uncomfortableness. So most of them felt like it was very niche. You'll see biomaterials are interesting but niche. And then they also found it like more valuable. In this case, you can see slightly more valuable than digital design approaches. Okay, so the physical component of it, even though they know it's very niche, they still found it very relevant. So, do we think that they're fundamentally opposed, digital design or material-driven design? No, not fundamentally opposed. I think it's interesting hearing the presentations today because so many people enter this from the perspective of digital fabrication as the entry point, but that's really hard to do with a class of 50 students, right? And it's beautiful work in the lab, but how do we get students engaged in it at scale to help them understand the practicality of it? And so I think it's some interesting questions there. I feel like my hidden slides are showing. Okay, yes, that's what's happening. Okay, I was like, I don't have all these slides here. Okay, the last thing then, in the context of faculty interviews, I had some really nice conversations with people. Like I said, faculty are really interested to talk about this and to have community around it. So I talked to a few different contexts. There's definitely a difference between bio-art and bio-design, and how sort of the cultures are emerging, subcultures are emerging around that in relation to the messiness factor and the grossness factor. I also found it interesting that I was sort of probing around this feeling of grossness, that many faculty were like, no, my students have never said that. And I was wondering, it made me wonder, why do my students say that so much? So I'm curious, if anyone wants to sign up for an interview, I'm curious of the experiences that you've had about this. This QR code is for the interviews, and then the next QR code will be for the survey. I'll show you in just a minute. So if you're interested in being interviewed, I would love to make this a more complex, and have both the faculty perspectives and the student perspectives of this issue in the research. And then also, I need more students to participate. I'm thinking about stalking them on Instagram if I can see if I can get their attention. But I have to lay out my strategy for how I'm going to get more student participation from here. But if you're interested in really getting involved in this and you want to share this with your students, I'm happy to invite more co-authors to the project. So that's all for me. Thank you.
Thank you very much, Zarin, for your insightful presentation. Communication is not just explain clearly some subject and some concept, but also to help people encountering unfamiliar situation, try to engage them, not just to resist to the experience, but to be engaged more. So thank you very much, Zarin, once again. So the second presentation moves from the classroom to co-creation setting, and it asks how regenerative principle can become something practical and shareable across participants from different backgrounds. So it's my pleasure to introduce Arthur Arayu, from the Universidad de Aveiro, presenting co-creating regeneration, regenerative strategic design principle to reimagine tourism in Portugal. It's a very different topic, but we are always in the same subject as the communication. Thank you.
Thank you. Hello, everyone. I'm Arthur Braga de Araújo. I'm from Brazil, and I'm here to present this ongoing experiment that's part of my PhD research. I'm investigating about participatory design as an approach for regenerative tourism development in coastal zones. And I will present a workshop that I facilitate during the Portuguese citizen science meeting that was last year. We know that in Portugal, tourism is at a turning point. We see from one side degenerative tourism creating pollution, different kinds of pressure in the environment. We see also cultural homogenization and social displacement. And we also can see, in some places, we can imagine and already see some regenerative seeds that also we can see in rural areas as community-led hosting, local market, and also valorization of nature and community wisdom. So my goal in this experiment is to understand how we can translate, how we can communicate regeneration without just using buzzwords. So I had the opportunity to facilitate a process in this meeting with researchers from different backgrounds, and my will it was to think about a practical way to translate regeneration. So we can, with participants from different areas, has a common background, common basis to make decisions, negotiate, and create possibilities to the future of tourism in Portugal, but also in other places. So this framework that I used, it was created by Caroline Tomás and Karine Freire. Both are researchers from Brazil, and they developed this framework that is part of my investigation of my state of art. This framework is based on three main principles, three main knowledge: system thinkings, ecofeminism, and buen vivir, or good living, bom viver. That is an Indian ancient knowledge that is based, that it was a worldview that values the harmony between nature and humans. So they created, from these three bases, 12 principles that includes relational, being relational, contextual, inclusive, equitable, adaptive, decolonial, and as you see, 12 principles that we use to guide this process that I facilitate during this meeting. So this workshop, as I mentioned before, happened in the National Citizen Science meeting with researchers, mainly women, from different backgrounds, law, biologists, and other interested parts. And our process, it was based on build trust. So we did a walk in pairs to try to connect these participants with each other, and also recall some bad and good experience that they had during trips around Portugal and other places. So they could bring together some experience and share this experience, this good and bad experience. Then we start to map what we called stones as challenges and tensions, and also seeds as good practice that they remember. And we map together to understand how terrorism works nowadays. And I present the 12 principles of regenerative strategic design to address a co-creation process where they had to use these principles to co-create possibilities for these experiences. The workshop was not only about generate ideas. It was a way to teach people how can we move from personal experiences to criteria, to tensions, and from criteria for proposals. So we did this process, and we had some results. We had 4 results in this group, and I will be very brief to explain the results. The first, it was Saber Tem Idade. It was an experience where seniors host small-scale local activities. The potential is clear limits and redistribution of visitor flows. The tension is fair pay and responsibility. The second, Conserva o Português, a small-scale program focused on local culture and products, shifting demand beyond big cities. The potential is local value retention. The tension is essentialism and governance, who defines authenticity in this experience, these experiences. The third, it was Na Periferia Está a Virtude, co-created roots with residents, mixing traditional and less conventional experiences. The potential is co-authorship and decentralization. And the tension is access and burden shift, moving pressures without care. The fourth and the last one, it was Tradição Tem Sabor, reusing degraded urban buildings as vertical ecosystems of crafts. Local commerce and a rooftop tavern. The potential is craft revival and local economies. The tension is touristification and rent pressure if governance is not built in. The key point here is how we can translate regeneration for the general audience. So we have some outputs of this work. The main output, the first output is the pedagogical device, when we can inform prompts, constraints, and safeguards. And the second output is the evaluation lens, when we read the workshop through mechanisms, trade-offs, and tensions, not only good intentions. And at the center, we have this learning loop showing regeneration as iterative process. Knowledge, decide, test, feedback, adapt, and generate more knowledge. Next, we propose 2 pathways. The first is participatory validation with communities. So this was facilitated in a controlled environment with researchers, with specialists, and we would like to create a tool to facilitate with communities. And also in education transfer, adapting this format also to inform new designers through design studios and tourism education, supported by a simple assessment rubric. This is an ongoing experiment, and our next step is to keep translating regenerative principles into practical way to inform, to communicate, to educate people about regeneration. And thank you very much.
Thank you very much for your presentation. Really interesting to see how the collaboration and communication among different disciplines can move from aspiration to real action, and how this can be communicated to different public, from general public, student education, and so on. Thank you again, Arthur, for your presentation. Now it's time to move to the third one, that is about sensory learning and collaborative design processes. And we now turn to a presentation that seems to focus on what connect disciplines, environment, and people, the human layer. It's my pleasure to invite Ines Meo-Blantar, I hope the pronunciation is not too bad, network strategist and creative knowledge transfer facilitator that assists innovation in communication, presenting between disciplines and the environment, the human layer. Please, the stage.
Thank you, Antonella. Thank you. Let me find my slides. I'm going to keep it short. So, basically, I'm a science communicator, but I think many of you have maybe different ideas where science communicators come from, like what they did before. So this actually doesn't focus on a specific research project. This focuses on like sort of conveying the power of science communication in projects like DESIGNAE to you, and also to showcase you some handles, some techniques one can apply. And starting with the first handle. This is actually a very personal handle. This is actually a tool we used at the Natural History Museum, like going into the conversation with kids' portraits of researchers. Just like to convey within like 5 seconds where this person is coming from, because in the end it's gonna keep a longer emotional angle, and people actually focus. And I'm revealing this to you, I'm showing you a childhood picture of mine. So where do I come from as a communicator? Indeed, I come from the southern Alps of Austria, a Slovenian-speaking region, funnily enough, close to Italy. And I fell in love with nature, I think around that age. I think it was pretty much that moment when I spotted that butterfly on the traditional dress that my parents put me in, and I just couldn't keep the eyes off this butterfly. And it stayed with me, this symbol of a butterfly is a later symbol for transformation that I actually then used in the course of many different things. But just like to give you an idea where science communication and knowledge transfer facilitation can come from, I then added up a degree in sustainable development by the University of Geneva. And before then I was basically working either in science or in communications, because it came to me quite early that like communication is a thing I really like here, to talk to people. I really actually love it, it fulfills me, and it's wonderful to build bridges. But science communication was not really existing at the time because it's a relatively new discipline. So I actually started to work in science communication, I would say around 15 years ago. And then moved into research, into different ecosystems. One thing that I basically always carried with me was my angle to human ethology, like the human layer. So I did study biology with a great passion for zoology, but I also basically discovered the human ethology per se as probably the most important tool in this project we are all like part of, because like in the all it's about convincing others to do the things, like how do communities work, how do we all get together, how do we trick ourselves, how can we work with senses, and Actually, I then had the wonderful chance to work with Pedro and build the innovation hub of the Natural History Museum in Vienna. And we'll come back to that in a bit. And one thing that also is interesting that I also have touch point with like deep science, deep research, biotech, and then very soft angles. And actually, I think it's important for me to have like all these years of background, because only now I think I'm able to connect the dots. I think before I wouldn't have been able to do that. And more recently, everything has changed. I decided to show you also what these kind of shows to see like how science communication per se has been applied 20 years ago, and how it's applied today in different contexts. So the first image I'm showing you is basically the University of Vienna, because I came to Vienna from the countryside, a young girl exploring the city, research life, and I had then the chance to do field work in the Alpine Alps. And why did I have to do that? Because I worked on the DARPA project, which was really weird. My first job ever being paid by US tax money. So that felt really weird. But why was communication a thing at the time? Because we basically had the job to recruit 200 people in the Alps. This was people with families that had at least 5 generations. And we had to do that because basically we had to get them to donate samples, because we were exploring the connections between HLA genes. These are the immunocoding genes basically that are responsible for smell. And like to see if there's some microbial family properties. And the Americans, okay, was around the time of 9/11, they were interested in that. And they were funding this in the Austrian Alps. But that led me to a very beautiful thing, because we had to talk with the people. We had to like let them know, and that was before any ethical things were much more severe, like why to donate blood, like what is gonna happen with my research. So basically, all this time ago, like we were doing science communication before it was even a thing. Okay, then it became a thing. My personal life, then led me later to Geneva, which is also basically very important. So in Geneva, they have a wonderful place. It's called the Swiss Center for Affective Sciences. And basically, they do everything about emotions and how emotions guide everything. So they have designers in there, they have neuroscientists in there, philosophers, linguists, super interdisciplinary point. I had the great luck of being able to work there for a couple of years. And I think it was just important for me just to soak the matter, to get to know all these interdisciplinary exchanges, because again, in Austria, when I came back, it was not a thing. So I could at least bring that back. There, science communication was already integral, because it was, an interdisciplinary center. It was integral to everything. So we did exhibitions. We talked a lot to people. It was not considered, hey, communicator, write us a press release, but it was just like a really important pillar of the center per se. Returning back to Vienna, I then came into the biotech scene, where we worked with stem cells, very critical materials, so bioethics became a thing. When I started there, people were like, yeah, write me a press release so we get more views, and like PIs were very hard to work with, because basically it turned out they only want to get, I just quote them, better PhD students. And I was like, who are better PhD students? Yeah, people who come from good labs. So that led me to some things like, aha, very interesting. Funnily enough, within 5 years at this institute that started with this very deficitary model, like sort of science communications basically disseminating what we say is right, but had no like engagement with people whatsoever, because they worked with highly sensitive material, namely like human blastoids. These are basically synthetic embryos grown from iPS cells. So all of a sudden there is the need to do something ethical, and we did it. I'll show you some examples later. But it has grown in that sense. The awareness like really grew within 5 years. The next station that is mentioned here, I mean, there was basically for me the origin of science communication per se in natural history museums or in museums. So I think they bring a lot of value when it's about communicating with people, because they really know it. Like all the researchers working there, like Pedro, are fantastically skilled science communicators, and they also know how to listen and to integrate other people's perspective. As a next slide, I'll show you just some examples of what ways you can think of, like when communicating projects, etc. One thing already mentioned before, so I come from the emotion research, and then the SDGs, the social developmental goals, I've seen them a couple of times today, were a thing. And when I read them, I was like, there is no like the human layer consideration whatsoever. You have these goals all of a sudden, but nobody tells us how to work with them, who actually really get people to do them, like to work together, to collaborate, because we're humans after all. And I was getting a little bit intrigued how to do that, and I think that effective communications, like working with objects, working with fantastic images, getting people in awe, like really makes a change. I have a picture of Pedro here because like we used to work together for almost 5 years, and we still do. For one action, for example, we collectively destroyed corals just to glue them back together, to convey the concept of coral regeneration. There was a beautiful thing adopted by little kids as well as important stakeholders. Another thing one can easily do, like when conveying these sort of things, is going totally outside the box. So as I mentioned, I was in a quite deep tech place, and there was like very high impact research. Basically, every lab had to publish at least one publication in Cell Nature Science, otherwise they were in trouble. So that was a little bit the environment that was there. And instead of doing an annual report showcasing all that, I just like proposed, let's do an art and science book instead. And we did that. So we had, like, we paired up with the wonderful class of the Angewandte, the University of Applied Arts, and basically just turned highly molecular processes into abstract photo art. And we did that also in co-creation with the artists, so it was like great fun. It was a white card. So it was a beautiful book that's actually published. And 2 years ago, I was in Bilbao and I actually saw that they sell it still there in the Guggenheim Museum. So it really went out of the bubble. And this is something we really want to reach when we want to have this kind of impact, not to only stay in our community, but really to reach out. Another thing that's important when reaching out is like, you have topics, but you need to discuss it in really big settings. So one picture you see for stage situation, it actually was an auditorium of 2,000 people. It was one of the biggest theaters in Vienna at the time. And we had a big open public debate about like frontiers and limits of biotechnology, also related with stem cell research. There was about the designer babies, etc. So we invited not only the head of the Bioethics Commission, but also a science fiction writer to basically prop it up. And then nobody, like my boss didn't believe me. He said, no, you're not gonna have this place full, it's not possible. I told him, like, listen, like if we do it the right way, if you find the right channels, and it was full. It was full house, and like it was quite amazing. Something that was basically destined for formats like this are museum places. We had this discussion before, hey, we need to reopen, and basically I think the willingness is there all over the museum landscape. I think the problem is just that it's a problem of funds many oftentimes, resources, because resources in cultural institutions are even more scarce, because you have to devote a lot of time to research activities, but then you're still a cultural public institution, so that becomes very tricky. And one way to finance this, for example, was participation to Horizon Euro project, where we worked on the future of taxonomy. So it was all about saving taxonomies, raising awareness for biological taxonomy. And there, I mean, you see this space here that was for that purpose transformed into a high-level stakeholder forum where we invited CEOs, as well as one like pioneer in synthetic biology, as well as like conservation people, just to have this moderated dialogue for the commission. But at the same stage, one day before, you had little kids playing. So this is also like very important to really mix audience and to also like really know that you have one message and you can just really address it in multiple ways, and like you just have to keep on doing it. I mentioned before, like sort of I'm a human ethologist by training, and I just want to show you a few examples. And funnily enough, there's already touch points with some things I saw today. So one thing I like to mention is a lot of about like making things visible, making things that are hidden tangible. Emotions like are a very funny thing. So this, I don't know if any one of you knows that that's a heat map of bodily emotions that was already done in 2012 by Aalto University in Finland. And what they did is they asked people, in the beginning like 700 people, to draw on a body map where they feel which emotion. And so basically this is not thermal scans, even though they look like it. It's just like what people self-report, how they feel happy and where in their body. That was then replicated, I think, 10 years later with a sample size of 3,000, but then cross-cultural, and it was totally consistent. And this now creates a beautiful basis, for example, when we want to explore our relationships with natures, to make everything visual. For example, this gives us a handle for biomimicry, biofeedback, right? If something feels good, if your new cool algae outfit like feels good, you can actually also see it, right? And then one thing I still wanted to mention, even though it was mentioned already several times today, so this looks a little bit squished, but it's basically an installation by Julia Lohmann. And what she did is she took all the bacterial cultures of her skin and cultivated them to make it visible. So when we talk about, again, human-centric design, again, we're not just one being, we are more than one, but things like that and installations that just like really help us to make it tangible, to make it visible. And like DESIGNAE is such a powerful project, and like, I mean, already what I saw today, I think it's gonna be the highlight of my year, the gathering of people today. And it's so powerful that, like, also in terms of visual communication, in terms of sensory communication that was already mentioned today. Haptical things are one very important thing, because right now, you know, you have this cognitive embodiment that is getting lost because everything is like digital. So this going back to a more analog haptical world is also a very important tool. And actually, I was just thinking when hearing you, maybe we should have started in kindergarten, and maybe those kids who are not disgusted by touching should be the future bioengineers and biodesigners. And I really think we have to start thinking that way. One thing that I just want to show, because Pedro is gonna continue and he's gonna a little bit go more in depth. We did really try at DEC 50, that was the innovation space as the Natural History Museum, to work with the collections, to really like build the bridges to different disciplines. And it was really not easy. And it's still an ongoing process, so we're super happy to know now that there's a paper out, that Antonella is here. We're definitely going to talk and like exchange. But it's super difficult because like we need to have this common ground of understanding, and also like we need to clarify all these questions we discussed today. But I think the most important thing is that it happens, that its exchanges continue to happen, and that hope and aspiration also continues to happen. And in that sense, I'm going to close with this flag, and I wonder who knows this flag apart from Eleni. Anyone else? So there is actually a big communication movement already existing. I don't know who is acquainted with the book, The Ministry of the Future by, thank you, Kim Stanley Robinson. It's a wonderful utopic book about like how you can turn everything around, everything. And so this is the flag of a so-called literally movement called the solar punk movement. So what they, and what you see here is the rays of the sun, like sort of as a source of power and of aspiration and hope. And at the same time, it means we can still trigger it around and we can build our own things. And I think we should do that by really focusing on our superpowers as human, because we shouldn't forget that we are highly social animals. We just have to be a little bit machinated to do so. So there's all the tricks, we just have to find them and apply them. And also, we are totally highly adaptive species, and we totally forgot that. So yeah, let me close with this little solar punk manifest here. Happy to chat afterwards and happy to pass on to Pedro, who's gonna tell a bit more about the fantastic role of the Natural History Museum Vienna in that context, for example. Yeah.
Thank you so much, Ines, to sharing with us your vision, your experience, and the role of the museum in science communication, engaging a broader population, not just the scientific environment. So we will stay into the museum context, and now is the time of Pedro Frade from the Natural Museum of Austria. So please.
Thank you so much. All right, so this talk is the result of conversations we had back there at the Natural History Museum. So I'm co-authoring it also with Ines, who was running our innovation lab before, and also with the director of the museum, Katrin Folland. And we started a little bit the conversation, what can we do with our collections? How can our collections be useful for regenerative futures? So the title is a little bit from inspiration to what? Well, you all know natural history museums. We have collections, natural collections from the natural world to the humanities. If you haven't been there yet, come for a visit. This is just a little sketch of how we kind of position ourselves. We don't really understand how we can be useful most of the times, and we have a big discrepancy between the curators, what they think their collections could be useful for. So there's a lot of things to kind of coordinate there, to clarify. We want to be a place where there can be brainstorming, also with you, and there's an idea of organizing a workshop around that role of collections, of natural collections, for biodesign, for regenerative futures. So you will hear from us about this. I think we'll organize something. Maybe writing a review paper would be a good idea. Also being a place for hackathons where software developers can come together and tackle a question in place in the moment. Of course, we want to be a place where you can find some ideas, some inspiration, but we don't really know how to do this. Of course, upscaling and looking for some kind of monetary success for the museum, because after all, we are poorly funded. Right. Actually, I thought I was going to be talking in working group 1 because I'm a marine biologist. So now I switched to working group 1. I want to talk about corals, okay? And what I find fascinating about corals is that, they are animals that, with the power of light, they basically fix carbon into rock, right? So they have these amazing superpowers. But what I also find fascinating about corals is that they use polyps, so these are modules that are all the same, and they repeat these polyps over and over and over to form colonies, and these colonies show up in 1,000 different morphologies, 1,000 different ways. Of course, we use them as taxonomists to identify them, but we have troubles with corals because based on the same genotype, the same genotype can show up in a completely different way. So this plasticity, so this turning on and off of genes, depending on the environment where things are growing, kind of brings corals to being this kind of protean life forms that show up in different ways. And of course, this can give a lot of ideas for many things you guys work with in terms of design. Another thing that I wanted to mention here is that, this capacity to actually integrate and create ecosystems, right? So we often see organisms as kind of being shaped by natural selection, right? An organism is something that is being shaped by the physical powers of the world. But we often forget to see that organisms also shape natural selection, right? And actually these organisms, they form ecosystems, right? So they are the actual architects of our world, and they are the designers of our world. They form reefs that can be seen from outer space and on which 1/4 of all marine species live. And often we forget that the fact we exist is because they created all these resources, right, that we use and upon which we evolved. And I just want to give you one more example about corals, because this is a paper we published with a colleague from the Medical University in Vienna. And this is about the living together of organisms that live in colonies. Basically, we deciphered and we modeled a way by which corals exchange, communicate within colonies, between polyps. They have these systems of microcurrents on their surface, and actually they are quite stereotypical, and we think this can be a really nice model to, model flows of information, flows of resources, the living together in cities, or the way resources can move around the world. This slide is copied because I wanted to link it to Ines, and I thought I was going to be talking in the morning. But I just want to give you maybe 1 or 2 examples of how we think our collections can be useful. I give you, for example, the example, a phone call I had once from the European Space Agency. They were calling and saying, hey, can we come and look at your sea urchin collections? So sea urchins have these spines that are really strong, but also really light. So the idea of space travel, finding the best carbon incorporation way by which you can have strong and light structures. And I think in the future, we will have to reconstruct the ecosystems we lost. And I think a museum is a place where one could actually look at baselines, right? Looking at genetic functions that are perhaps already lost. And by looking at these collections that go back to the 18th century, for example, we can probably learn a lot about that. I want to also put here together maybe just two more notes, if you allow me, maybe for the people who want to maybe eat insects. If you visit the taxonomists in the museum, they will tell you that insects are actually diverged from crustaceans. So actually insects are crustaceans, okay? And so you will be basically eating something like a lobster. And I think a lot of people like lobsters. So there's one reason. And then, yeah, I think I'll leave it there. And so one idea we have, and together with the director, is that, so we, as you know well, we have a lot of exhibitions. There's running exhibitions. These are just some of the examples of exhibitions we have now. Glaciers out of balance, but also ways of producing food sources that are kind of out of the box and restorative as well. So one thing we want to mention is that we want to have, in 2029, an exhibition about bioinnovation. So this is something about bioinspiration, products and outcomes, and we would actually invite you guys, and we can think of the best way to do this, that some of the outputs of this project could then be shown at the Natural History Museum in our exhibition. And I think with that, I finish, and thank you for the opportunity. Yes.
Thank you so much, Pedro. Very interesting. I really like the proposal. Can be also something that can be moved from different museums so that many people can access to the same. So this is the end of the session that was dedicated to communication, to dissemination tools, to training framework. The speaker show us how to make accessible interdisciplinary ideas to a broader audience. And I would like to thank you very much to share different perspectives, but highly complementary. And very inspiring also for our meeting tomorrow afternoon in the group. So we have many ideas on the table. So I would like to take the opportunity to thank Nina and the team for the work they are doing. That is huge. It's a great work they're doing for these days. This is my opinion, but for me, it was a very nice day. I learned a lot. Very inspiring and open-mind day, because we are in the same room and we are different expertise, but we are able to communicate, to share the experience, and we can learn from other experience, bringing ideas in our field. So thank you, Nina. I really appreciate it.
Thank you. Thank you so much, Professor Antonella. Thank you to all the speakers. Thank you. So without further ado, I'm going to close today's session. We have the dinner already booked in half an hour or 20 minutes or so, so we're just Very quick refreshment and then let's go. I will meet you again tomorrow. We have, so the first part of the morning is dedicated to the management committee meeting. And then please remember the schedule for the buses. I will text you again via WhatsApp so that everyone is reminded of location and time. If you're not part of the management committee and you're not part of the core group, your bus will, you only have, you will join us at 11, which is when the bus passes. So I hope you enjoyed today. It was a very long day, but with lots of different insights from people from very diverse disciplinary backgrounds, which is very enriching. Even, I mean, for me it was mind-blowing. So I'm going to have to also absorb all the insights, all this information, so that I'm able also to rethink about the plan with my fellow colleagues, to make sure that we take full advantage of DESIGNAE COST Action. So thank you so much for sticking around, and I will see you tomorrow. Thank you. Thank you.